Right, so a very good morning to all of you, and welcome to APA India 2020 Virtual Conference Webinar Series. If Patanjali, if you can just confirm if my voice is audible.
Can you hear me well?
Yes, I can hear you. Is my voice audible,
Yeah.
Patanjali?
Yep.
Okay, great. Thank you. So, very good morning to all of you. Webinar number 13. The monthly theme for the month of October is biotherapeutics development. So all the webinars in discovery, biotransformation, regulated bioanalysis, and essentially the CMC would be aligned to biotherapeutics topics and theme only. So today is regulated bioanalysis day, 9th of October, and webinar number 13. So before we start, just a few slides on introductory slides on APA. So special thanks to all our organizers, all the presiding chair-elect for their untiring efforts to bring in such an interesting agenda on board. So a big thank you to all the presiding chair and chair-elect of committees, namely regulated bioanalysis, discovery, biotransformation, and chemistry, manufacturing, and control, that's CMC. A special thank you to all our organizers as well, because organizers plays an important role. The president, chair, and chair-elect, they're supported by a very active team of organizers, organizing committee members. So a big thank you to all of them, because as you can see, they are all from different world-renowned reputed CROs and industry and academia, and they're really busy, but still they can spare some valuable time and insight into the organization of this APA India chapter. So big thank you to all our organizers, the Committee for Regulatory Bioanalysis, Discovery Bioanalysis, for Biotransformation, and for CMC. Again, very big thank you to all our sponsors, Shimadzu, Science, Zeph, Sai, Phenomenix, Thermo Fisher, Agilent, Syngene, USP, NATCO, Vida Clinical Research, Bristol Myers Squibb, Zoetis, Nanotemper, IVAL, In Vitro Met Labs, Origin, Vuxi Aptec, and Loris Infosystems. So a big thank you to all our sponsors for their support, not only for APA India 2021, but for their consistent support since the inception of APA India chapter. I think most of the sponsors are associated with APA India chapter since 2009. So a big thank you to all our sponsors. This year we have collaborated with the Ministry of Skill Development and Entrepreneurship, a council called Life Sciences Sector Skill Development Council, whose major mandate from the government is to create an ecosystem where industry-ready candidates can be made available to the industry. So they work closely with academia, pan-India, and also with the industries. So we have collaborated with Life Sciences Sector Skill Development Council also. So thank you to LSSDC. A couple of very quick announcements about the APA India 2021 poster session. As all of the poster session poster presenters are aware that we were supposed to do this on 2nd of October. Since we received a lot of requests from the poster presenters that they need more time to prepare their presentation. So we have rescheduled this poster session, and now it will happen on 13th of November. Initially, we thought it can happen on 6th of November, but because of Deepavali holidays, Deepavali is on 4th, so we may have, people taking vacations, and there could be an issue for many people to attend. So we have rescheduled it to 13th of November. So all those poster presenters whose abstracts have been shortlisted and who have received email confirmation from Boston International, keep your presentations ready, and the final poster session will happen on 13th of November. Each of you will get 8 minutes of slot. That is essentially 6 minutes for presentation and 2 minutes for Q&A. So in total, out of all those shortlisted posters who will participate on 13th of November in the poster session, 9 poster winners would be selected by the panel of judges, and there will be 3 best poster winners for first prize. Same way, 3 best poster winners as second prize and 3 best poster winners for third prize. So there are very interesting cash rewards. There'll be a memento, a certificate, and some surprise prizes as well for the winners. So just would like to wish all the poster presenters very best of luck for their presentation on 13th of November. Also, at the end of every webinar, till now we have conducted around 12 webinars, and we have always done a poll with few questions about the webinar, and all those who participates in that, they become eligible for the Kindle draw, which we do at the end of the webinar. So would like to congratulate to all of our winners. I think I'm yet to include the winners from the last webinar, webinar 12. But would like to congratulate all our winners and again urge all the participants to please participate in the webinar poll at the end of the webinar. For Q&A for participants who are joining for the first time, since as a participant, as an attendee, your audio and videos are disabled. So if you have any questions for any of the speakers, feel free to use the Q&A tab at the bottom of your screen to post your questions. Even in chat box, you can post your questions. After the talk, our moderator will take up those questions with the speakers. And again, speakers would be available in the panel discussion at the end of the webinar. So even during that time also, you can get your doubts clarified. And during the webinar, if you're not able to answer or address all your questions and queries, then would request you to post your questions on bostondb21@gmail.com. And we will reach out to the concerned speaker and get your doubts clarified. Once again, do participate in the poll at the end of the webinar. Only then you'll be eligible for the Kindle prize draw, which happens at the end. The webinar talks, all the webinar talks will be uploaded on our YouTube channel of Boston International after having due consent from the speakers. So would request all of you to just keep or do subscribe Boston International channel as well. In the month of November, all the talks, all the webinars will be uploaded on this channel. So you can always go back and refer any of the talks for learning purposes. So with that, without further ado, let me invite the session chair, Patanjali, for his introductory remark, and then to start the session with introduction of our first speaker. Over to you, Patanjali. Thank you.
Can you hear me? I'm audible?
Yes.
Okay.
Yes, sir.
Good morning, good afternoon, and good evening, wherever you are. This is Patanjali Kadiala from Bristol Myers Squibb. I've been with Bristol Myers Squibb for the last 29 years and currently working as Associate Director, Outsourcing Operations Management Group. I'm happy to initiate this webinar for today, the theme being biotherapeutics development. And you might know that if you guys know, the insulin was discovered 100 years ago. I just was Googling just a while ago, 1921 by Frederick Banting. I'm not sure whether anybody is celebrating 100 years of insulin discovery or not, but since then, so much technology has evolved for the treatment of diabetics in terms of different types of insulins and different types of delivery materials, like the syringes to the pens and the pumps for these juvenile diabetes patients. So much has evolved through this insulin. Similarly, the promising field of biotherapeutic medicines has gone a long way in delivering a new generation of treatments known as biotherapeutics, also called biotech drugs or biologics, which are made from living organisms, harnessing living cells to make or modify protein products to create therapies. What is so special about biotherapeutics? Their manufacturing process is very complex. They're modified to ensure they function as intended. The most effective cell line is selected for expansion and is grown in bioreactors and carefully monitored. And they require highly sophisticated production control and regulatory processes to ensure their quality, safety, and efficacy. And to support all these things, it comes into the play the pivotal role played by the bioanalytical assays for the PK and AD and NAB. And that's where the bioanalytical scientists come into picture, where their assays are the key cornerstone or play the pivotal role to provide a data which is applied for the safety and efficacy of the patients and the subjects. And that's where the real data starts and help with the discovering new medicines for the patients. With that introduction, I'm going to introduce Darshana Jani, who's going to be today in a recorded version, and unfortunately she cannot participate live. Darshana Jani is the Director of Bioanalytical and Biomarker Development at GenoBio USA. Prior to this, she was Scientific Associate Director at Pfizer, Cambridge, where her role was to serve as scientific and technical lead for both CROs and internal teams for development, validation, and application of bioanalytical assays to comply with scientific and regulatory requirements. Darshana has over 25 years of experience supporting preclinical to clinical studies, holding positions with Sanofi, MedImmune, Biogen, and Pfizer. Darshana has risen from ranks at the outset, developing and applying bioanalytical methods while assuming numerous responsibilities germane to regulatory compliance of the validation and application of group procedures. She has guided bioanalytical lab groups, held responsible for overseeing and all phases of drug discovery and development, a task which incorporated product characterization, potency determination, immunogenicity testing, as well as surrogate biomarker assay development, validation, and sample testing. Darshana has published several recommendation white papers with the industry, academic and regulatory peers in the area of biomarkers, as well as immunogenicity. Right now, I request Deepak to start the recorded version of Darshana Jani.
My thoughts on the bioanalytical driven. I am Darshana Jani from HNS Bio, located in Lexington, Massachusetts. I will present my thoughts on the bioanalytical strategies for bispecific antibodies in clinical development. I have been in the field of bioanalysis for last 25 years, supporting immunogenicity, PK, and biomarker analysis. Prior to joining Agenus, I have worked at Pfizer, Biogen, Sanofi, and MedImmune. Due to the complex structure and complex mechanism of action of bispecific antibodies, innovative bioanalytical strategies may be required for PK and immunogenicity of this particular class of therapeutics. I will start with a brief overview of bispecific antibody therapeutics, followed by my thoughts on bioanalytical assessment, covering immunogenicity, PK, and soluble targets. I will wrap up with the key bioanalytical considerations. What are these bispecific antibody therapeutics? They are large family of molecules designed to recognize two different epitopes or antigens. On the right, I have shown various forms of bispecific antibodies. I copied them from the public domain. Bispecific antibodies come in many formats, ranging from relatively small proteins, merely consisting of two linked antigen binding fragments, to large immunoglobulin G-like molecules with the additional domains attached. They are designed to provide targeted therapy, making them the attractive candidates for especially oncology type of conditions. If we look at the approved bispecific antibody therapeutics, in year 2009, Removib was approved. It is a trifunctional IgG-like structure, and it binds to EpCAM and CD3, used for malignant ascites. Then FDA approved in 2014 Blincyto, which binds to CD19 and CD3, but it has a very different structure. And the third bispecific antibody is Hemlibra, which is a humanized IgG4 used for hemophilia A. So now we will talk about the bioanalytical strategies for these bispecific antibodies. So first question comes to the mind is, what are the key drivers and how we can effectively implement bioanalytical strategy. So there are four main areas. First is the regulatory expectation. Second is structure of the drug and target. Third is mode of action and disease area. And fourth, I have covered here the sample collection. So all these four items together really drives the bioanalytical strategy for immunogenicity assays, PK assays, PD assays for cytokines or targets, and the reagent support for the support of all this bioanalysis. So starting with our first regulatory expectation, in the public domain, I could find that FDA has published last year bispecific antibody drug development guidance. The key point for this guidance is that it focuses on two major classes of bispecific antibodies, whether the therapeutic simultaneously binds the target or not. The bispecific antibody development program is subject to all pertinent laws of biologics. Bioanalytically, the guidance talks about developing and validating PK assays, which are pharmacologically relevant to PK/PD assessment. And immunogenicity is also been mentioned here. Immunogenicity may be of concern because of the novel epitopes in the unique structure of these antibody molecules. So overall, bispecific antibody assays are used for determining and supporting safety and efficacy of the therapeutic. The unique structure of the molecules may require modification in the traditional analytical strategy. The molecule is heterogeneous in nature. The size can vary from single chain to multiple chains, as we looked at it earlier. So the bioanalytical platforms which can support bioanalysis can range from traditional ligand binding assays to cell-based assays like flow cytometry. And in addition, multiple assays may be needed for immunogenicity, PK, and PD analysis. Understanding the stage of the drug development is also necessary. It determines what is the goal of the program, what is the goal of the study. The majority of the times, we want to understand the exposure-response relationship for safety and efficacy. So overall, if we start with the immunogenicity first, what are some concerns for antibodies in the clinic? So these concerns are not any way different from biologics. So the first is the pharmacokinetics, decrease in concentration or increase in clearance. The second concern is pharmacodynamics, lowering of the efficacy of the therapeutic, loss of drug effect, shortened duration of the response, cross-reactivity to endogenous protein. And the antibodies can trigger or induce hypersensitivity or anaphylactic reaction or injection site reaction. And with all being said, there might be the cases where despite of antibody generation to the molecule, no impact is noticed. So with that in mind, if we look at the immunogenicity assessment roadmap, the first two factors which really impact the overall immunogenicity strategy is the risk evaluation and the availability of the reagents. And then that will determine the assay and platform selection, followed by validation and sample testing. And of course, the bioanalysis is not complete until we look at the PK/PD relationship. So little bit looking deeper into these parameters, that risk evaluation and reagent category, we are aware of patient-related and product-related factors. There are multiple white papers in the public domain which can help us understand how to assess risk evaluation. And for the reagents, we are all experienced with the biologics. However, with the bispecifics, with its unique structure, with its unique structural properties, little bit more time and resources are required. For product-related factors, same thing again, its unique structure. Then in step 2, platform selection, and whether to use a multi-tiered strategy or to develop unique assays based on the structure is very important. Considering the therapeutic area, the disease condition the therapeutic is used for, so metric selectivity and cut point selection is also the important consideration. Then once we go into the assay validation, the assay is still a biological method, and if we need to follow the current regulatory documents. For the unique assays, we can have the scientific judgment in addition to what is in the guidance. And for the sample testing, for immunogenicity particularly, many times the one point which we all need to be always think about is the in-study cut point, if warranted. So the gold standard immunogenicity testing by the tiered approach is well known. The samples, whether they are pre-treated or not, but samples are first tested in the screening assay using statistical cut point showing 5% false positive rate. ADA positive samples are then tested in the confirmatory assay using the whole drug. And here again, the statistical cut point of 1% false positive rate is applied. And the results are expressed as a titer after sample is titered in the titer assay. Now, the key component of the bispecific antibody therapeutic is if the confirmatory assay is positive for a sample, then we want to test the antibody specificity using the domain characterization assay, which is the specificity assay, whether the specificity is towards the arm 1 or arm 2. And similarly for the neutralizing antibody assay, whether the neutralizing antibody potential is toward a specific molecule, specific part of the molecule, arm 1, arm 2. So to better understand that, I have used as a typical of a bispecific IgG-like antibody molecule, which has one arm is specific to target 1, second arm is specific to target 2. And using a gold standard MSD bridging assay platform, where biotinylated bispecific antibody was coated on the streptavidin plate, and the anti-drug antibodies were detected using ruthenium labeled whole molecule and getting the signal. Here, this part of the bridging assay is very similar for any biologic. But then when it comes to the cut points, we need to establish 3 confirmatory cut points. One is towards the whole construct, second is towards the target 1 arm, and third is towards target 2 arm. And that's where the domain characterization really plays a big role during the screening portion. Now, if we schematically look at this confirmatory assay versus domain-specific assays, the first on the left cartoon is of the screening assay format, which I showed earlier. Now, in the middle is the confirmatory assay format, where the sample is incubated with the whole drug. And if the antibody is specific to any part of the molecule, it will bind to the drug, and we will see the reduction of signal. Now, if the sample is antibody positive, then we want to further determine where antibody binds to, whether one arm is more immunogenic than the other, whether antibody binds to a special portion of the molecule. And here I have shown the two arms of the molecules, that the samples are incubated with either one separately in the two separate assays, and we determine the domain specificity. This portion is very important when we really need to interpret the immunogenicity data. Now I will little bit talk about the neutralizing antibody assay. If the sample is positive in the confirmatory assay, we want to check the neutralizing potential of these antibodies. Now, in that case, domain specificity comes in the picture. If we find that the antibodies are against one arm of the molecule, and if that arm has an endogenous counterpart, then it is very important for the safety that we measure the neutralizing antibody potential. Understanding the domain specificity may be more predictive of the neutralization potential of ADA than the NAB assay itself with the poor sensitivity or drug tolerance. So my thought is that the NAB assay and the domain characterization of the ADA assay should be complementary. We face a lot of challenges with the sensitivity, drug tolerance, matrix interference in the neutralizing antibody assays. But if we have the domain characterization, it helps us to interpret the data. So overall interpretation of immunogenicity data, it requires comprehensive assessment. It is not just showing presence or absence of anti-drug antibodies and their neutralizing potential, but they should be the impact should be assessed comprehensively, starting with the onset of immune response and what is the kinetics, what is the impact on the drug exposure on the PK and PD drug activity. And is there any impact, any relationship with the adverse events? We typically look at the immunogenicity assessment in this way for any other biologics, and bispecific antibodies are no exception to that. With that, now I will go into the typical PK strategy and what are our key considerations. Always our first question is, what is the appropriate assay format selection? What do we want to measure? Are we measuring active? Are we measuring total? Are we measuring free? So on the right, I have a cartoon. Sorry, the reference is not shown here. I don't know why it didn't show up, but this cartoon is from Yoming and Vibha's paper published in 2014 in the Bioanalysis journal. And it very well depicts that free assay, which is the unbound drug assay, what kind of assay reagents you will use. The second point to consider in the PK assay is to implement sample treatment to overcome matrix interference or not. And the third is really a lot of PK assay platform, like PK assay development, depends on the reagent production, which is fit for purpose. Here I am showing the selection of appropriate assay format, especially for bispecific antibodies. We have seen earlier in the guidance, FDA guidance, that pharmacologically relevant. So we want to ask the question, are we looking for bioavailability or are we looking for exposure? And based on that, we can develop the assay either to determine free or intact molecule or to determine the total molecule. So I have shown the reference below on the left. So this scheme mainly has been adapted from that reference. So if we want to measure free or intact, we can have a functional bioassay. We can have a bifunctional ligand binding assay. If we want to measure the total to address the questions for exposure, we can have total ligand binding assay or monofunctional assay, or we can have more than one ligand binding assay. And if need be, we can use hybrid LCMS for intact or total PK determination. Now, if we look at if we look at the gold standard, our ligand binding assay ligand binding PK assay, where we use anti-EAD for target 1 as a capture, and the second anti-EAD, which is labeled with ruthenium labeled as a detector, we can measure the full molecule. This LBF assay format has some limitation. This format is prone to the matrix interference, and especially if there is abundant amount of the target circulating. As for example, the drug is not captured when the antibody binding site is blocked by the target, which is the middle figure. And then if we look at the right, drug is not detected if the antibody binding site is blocked by the target. So PK assay format is prone to matrix interference, and the impact of matrix interference is really severe. It can underestimate the drug, which ultimately provides inaccurate PK profile and ultimately impact on the dose calculation and the safety concern. In order to mitigate, there is ACE treatment, that is spade treatment, where you use the beads, use of specific and non-specific blockers in acid diluents, as for example, anti-idiotypic antibody for target blocking. And ultimately, if nothing is useful, then we may look into changing the assay format from ligand binding assay to the hybrid LCMS format. So at this time, I also want to touch base a little bit on the critical reagents. For bispecific antibodies, creating anti-idiotypic antibodies is necessary. Labeling the drug for the capture and detection really requires lot-to-lot bridging and lot-to-lot maintenance based on the molecular structure. So it is very important that the process is established for bridging the lots early in the assay development. Well-characterized reagents are also required, whether they are characterized at CRO or in-house where the assay is developed or where the assay is validated. It really doesn't matter where the characterization happens, but early assessment of cross-reactivity with the matrix components using these reagents is important to characterize issues after the storage. And the biophysical characterization to understand the aggregation of the molecule, to understand other physical characteristics of the molecules. You can get some of these characteristics from your CMC team, but once the reagents are labeled, then it becomes the bioanalytical issue and important to characterize the reagents. I have summarized the reagent planning for the bioanalytical assays, which can be applied to any biologics, including bispecific antibodies. The reagents which I have listed here are specifically for bispecific antibodies. The point of the slide here is that the reagents generation should start early. About 6 to 8 months prior to the preclinical stage in order to characterize all the reagents. If the neutralizing antibody assay is the cell-based assay, then important to identify the cell lines early in the game. And the scale-up of the reagents can happen during the clinical phase after the IND is submitted. So the reagent planning and reagent generation should also be an important driver in any bioanalytical strategy. If we look at overall bioanalytical challenges for bispecific antibodies, starting with immunogenicity, based on the complex structure and the novel epitopes which might have been introduced during protein engineering of the molecule, it will drive the immunogenicity strategy. I want to point out here that in the public domain, what I have read so far, there are 2 antibodies which are globally approved. Rimonabant had 0% immunogenicity rate, which was IgG-like molecule with no ADA incidence at all. The second molecule, Blincyto, which is not IgG-like molecule, it is just 2 single chain, and that has 1% antibody incidence. So immunogenicity incidence is low. However, complete bioanalysis is important for the domain characterization for a sound immunogenicity strategy. And matrix interference, drug tolerance, target interference, these are all the different challenges we really face during immunogenicity assay development. In PK, the question of free and total, sometimes very early in the assay development, the question is not well understood. The sensitivity and the availability of the reagents. For the target measurement, for the target assay development, many times ultra low measurements are required and the appropriate format needs to be selected. And more than one target assays are required. And for the structure itself, which impacts the stability of the molecule, and understanding the biotransformation is also very important to understand the challenges of the bioanalysis. I will now like to wrap up with this summary slide, that what are some key bioanalytical considerations for this particular bispecific antibody therapeutics. So risk assessment is essential for immunogenicity strategy. The reagent generation is very laborious and expensive. It is really important to start development. It is important that technical and scientific integrity is maintained. And at the same time, we need to be open to the innovative formats depending on the molecular structure. During the validation, typically we follow the current regulatory documents, but sometimes there is a need to scientifically mold the validation based on the unique assay format. And then the clinical relevance of the immunogenicity data and the PK data. So important that the open dialogue is maintained with all stakeholders for the information exchange. So with that, I hope I have provided some insight into bioanalysis of bispecific antibodies, and it will be useful to all of you who are listening. Thank you all for listening me, and we are ready for question answer. Thank you.
Deepak, this is a virtual recorded version, so I would like you to present the speaker participation certificate for Darshan and Jani. Okay. Thank you. And then I would like to invite Chef Ali Gupta to introduce Aparna Kasinath, head of Syngene Bioanalytical Lab. Chef Ali.
Yeah. Thanks, Patanjali. Yes. So Aparna Kashinath, she's just has been a friend. Just hold on. Just for over and has been with Syngene for over 19 years. Been working on drug development, bioanalytical assay, and has bioanalytical assay experience, which includes for over a decade of experience in immunoassay development, transfer, validation, and sample analysis in support of PK and immunogenicity studies for novel biologics and biosimilars. She's currently the head and test facility management for the lab. Aparna is a PhD from Sardar Patel University, Gujarat, India, and was selected as the UNESCO Fellow of Czech Academy of Sciences, Prague, as a part of her PhD program. Aparna has co-authored several recommendation papers in AAPS journals with global key opinion leaders on the topics of PK, immunogenicity, and NAB assays. These include recommendations for development and validation of immunogenicity assays in support of biosimilar programs, AAPS journal, systematic verification of bioanalytical similarity between a biosimilar and reference biotherapeutic, committee recommendations for development and validation of a single ligand binding assay to support PK assessments, and recommendations for development and validation of NAB assays in support of biosimilar assessments. She is also an active member of various global scientific groups and works to not only synchronize bioanalytical practices with global regulatory requirements, but also strives to bring out the Indian bioanalytical perspective onto a global platform for effective harmonization. Aparna is responsible for GLP of her lab to ensure scientific and regulatory compliance of data generated. In response to the corona pandemic, Aparna and her team were instrumental in setting up one of the earliest COVID-19 RT-PCR testing labs. Additionally, her team has contributed with clinical validation data for subsequent commercialization of S1 IgG serology kit. This team continues developing and validating assays for SARS-CoV-2 specific assays for biologics and vaccines. So it is a pleasure to have Aparna Kashinath today to, to share her views with us. Yeah, please, Aparna.
Thank you very much, Shefali, for the introduction. I'm going to quickly share my screen. Yes. Could you confirm you're able to see my screen?
Yes, very much. Go ahead. You're ready to go.
Thank you. Thank you very much. I'm going to speak about a topic that I'm sure is very close to all your hearts, all our hearts actually, about really how we managed bioanalysis during the COVID-19 pandemic, and especially if we were a regulated laboratory, how was it really done? So Syngene's large molecule bioanalytical laboratory works on various different types of immunoassays to support vaccines as well as biotherapeutics. And the work that we do is to be able to support with data for safety and efficacy endpoints for early as well as late phase trials. So there's a lot of patient trials and a lot of patient data that comes out of our lab. Now, to support these trials, we use the traditional as well as functional cell-based ligand binding assays as well. So here, the ligand binding assays that we use are for safety and efficacy measurement. We're able to, we use multiplexing as well as singleplex assays, and the assays that are developed both for vaccines as well as biotherapeutics, as per not only regulatory requirement but as per fit for purpose, for a certain evaluation of a trial. These assays are developed to be matrix specific and drug specific methods and of course validated for their internal use. And when it comes to compliance, the assays that are developed by this laboratory are OECD GLP compliant, as well as WHO GCLP compliant in terms of the studies. And of course, the platforms that we use are ELISA. We use a lot of mesoscale discovery, or the MSD ECL, a lot of functional cell-based assays, the Alpha ELISA platform, and automation is via TCAN. So this way, the laboratory supports PK, immunogenicity, as well as PD data that comes out that is needed for safety and efficacy measurement. Now, the assays that are run by the laboratory are designed based on the molecule. So the thing about large molecule is, these are proteins, and these are proteins that are coming with so many different types of structures, so many different types of epitopes and various different targets. So it is really important to design these assays based on the molecule. And then these assays have to also talk to the target, definitely have to be taking the a population, the disease, and of course the intervention, and at last but not the least, the expected therapeutic presentation of the molecule is something that is that has to be studied while designing and validating this assay. And of course, the drug mechanism of action, as you can see here on the screen, is something that we will have to stringently worry about in the beginning, and then validate and start getting comfortable about when we are really developing and designing large molecule assays for various different diseases. Now, the challenge with large molecule assays, which have to be so specific, which have to be so target based, is that these are always multi-step, and they often involve an elaborate sample pre-treatment, and these pre-treatments are usually done to remove interfering substances or proteins or targets, as well as circulating drugs. And this really takes place before the actual analysis. So be it a singleplex or be it a multiplex, or it could be a cell-based assay, sample treatment, elaborate steps, multi-steps is something that is mandated to get a fit for purpose assay in a large molecule environment. So what really happens is that these assays therefore become very skilled and as well as skilled people intensive. And of course, they're dependent on high-end technology for accurate, sensitive, selective data acquisition. And then we use, again, technology as well as software for regression of these of the data that we obtain. And then, of course, these studies are always run under stringent GCLP and GLP compliance, as well as SOPs that are set up in the laboratory. Now, we have been doing, this laboratory has been functioning, and we have been doing this for more than a decade. I would say about 14 or 15 years now. So in 15 years, we had set up a mechanism, we had set up our systems and processes, we had staff trained, we were really in a happy place. Our systems were mature, our processes were functioning as they should for uninterrupted data as well as study delivery. And then really the unexpected happened. So when the COVID-19 pandemic struck us in February 2019, we were really forced to what we call reimagine and quickly adapt or transition to new ways of working, new ways of ensuring that drug development support does not pause, because we knew there were patients that were waiting for critical medication, and that medication could only be approved if the data from our laboratory went on time. Therefore, we really could not stop, or we really could not pause. So since the bioanalytical services department, art, science really is considered as an essential in the drug development and the healthcare infrastructure, so we were lucky that through the lockdown, we could remain open and operational in order to support all our clients and manufacturers that we had collaborated and we had contracted and who had contracted with us. So which also meant that we had to really, on the feet really think, and had to switch to unique solutions for our systems and processes, for our people, and all this without really compromising an inch of quality or integrity, and of course the GLP compliance of every single number and data that we generate in the laboratory. And of course, our first priority was the safety of our employees and our contractors. So we evolved. So to reduce employee density at site, we moved to work from home for staff that do not usually work on the bench. And then we implemented a shift or a staggered system for staff that need to really work on the bench, and in order to reduce the density of the laboratory as well as campus at any given point of time. And then there were zones that were implemented, so that we are able to have our analysts and our study directors all stay in their own zones. So the physical interaction and the physical distancing is really conserved, and physical interaction is reduced. It's sad but true that we had to reduce physical and face-to-face interactions. And in our laboratories, we modified our labs in order to bring in seating as well as bench arrangements. So we have staggered bench usage. And then of course, there was food courts and satellite zones, and then there was this very strict COVID protocol that we had to follow, in the laboratory and across the campus, where staff were provided with PPE. And then there was this constant messaging about testing, tracking, about sanitization, masking, gloving, hand washing, etc. That really went all the went over these messagings went over again and again, to reduce the chances of spread. And at last but not the least, we also moved to an early detection as well as isolation. So we mandatorily monitored temperature twice a day for all our employees, and we ran routine testing by RT-PCR in our own laboratories for our employees. And of course, as soon as the vaccines came out, all that were eligible were vaccinated here at Syngene. And next, to really achieve a GLP compliant operational rhythm, there were certain roles and responsibilities that we had to consider for continuity. So there was a role of the test facility management, and then the study director, the study personnel, the document reviewer, at last but not the least, the quality assurance. So along with these roles, we knew that these are really the critical aspects of a GLP study, where all the way from the test facility being the overall responsible for the functioning of the laboratory, to the study director who's overall responsible for the conduct of the study, to the study personnel who's overall responsible for the experiments that he or she is doing, and then the document reviewer that is responsible for the review of each and every data and document that goes out of the laboratory, and of course a quality assurance program on team that are responsible for the quality of not only the studies but also the facility. So these critical roles really had to be thought about, because we could not really pause or cease doing any of these roles or performing our studies based on GLP. So what did we do? Keeping in mind the safety of these roles, we really transitioned the way we worked, and we kept continuing bioanalysis for all our studies. So when it came to the test facility management, we adopted a hybrid working system where I could either be on site or I could be remote, and I could still be able to monitor, and I was still able to monitor really what's happening in my laboratory. But in terms of the study director roles, most of the study directors were mostly on site on alternate days or on a shift basis, with a few exceptions. When they had plans and reports to write, then they would do it from home. But otherwise, we made arrangements so study directors could really stagger and come to work, but be on site for the conduct of their studies. When it came to the study personnel or the analysts, again, we adopted a staggered model where, with social distancing and of course regular COVID-19 protocols, and the days when there was a particular study that was running, the study director and the study personnel were always present, and there was always a document reviewer that was present to make sure that things are working well. So for the document reviewers, we had a hybrid facility, hybrid working. As I said, for facility reviews, they would do hybrid, for facility reviews, they would do on-site for study and facility reviews, and then they would do remote reviews for electronic as well as scanned paper data. So that's one thing that we changed. So these annexures that we worked were on, the study directors would, the study personnel would fill up the annexures, the study director would review the annexures and then would scan it to what we call as a document review or QC team. And they would sit at home and review a scanned paper data and give their comments on how the data is trending or how the study was really performing with respect to QC. Now, the quality assurance also adopted a hybrid system, but if at a given point of time, there was always one set of team that was present on site, and there was another set of team that was working remote continuously through the entire pandemic. Now, our study directors, as I said, had adopted an agile process of study conduct and review. So they kept on doing their work on a daily and a weekly basis. On a daily basis, the scheduling, the plate mapping, work list generation, procedure generation, then discussion with analysts, then looking at verification of assay requirements. All this was done in person, and if there were additional conversations to be had, it was done on Teams. Then when it comes to when it came to the review of results and ISR, and if there were deviations, there were OSs, there were troubleshooting to be done. This was also done as much as possible in person to ensure that challenges and operational and scientific alignment were handled in person. When there were non-interactive activities, these were done either on emails or via MS Teams. And of course, we continued our chats with and talks and our updates to our sponsors weekly. So we reviewed data conclusions and hypotheses. We ensured that critical path alignment was always there, and then we addressed all current issues. And this was done on MS Teams. And every single call, we also gave a COVID-19 update to our sponsors and kind of made them comfortable, saying, "Don't worry, we think we're going to look after your study. We'll do everything we can to make sure we are not able to cross deadlines." So we did the same thing for the data reviewer, as you can see on the slide. The data reviewers came in daily, and their work with regard to facility or study documentation or SOP and procedures was really a daily job. So the team worked in staggered shifts, but there was always a 30 to 50% of the team always available on site, and they also adopted the online review via VPN, etc. When it comes to the quality assurance team, it was a little more important that the quality assurance team really does not deviate from its assurance SOPs. Therefore, for inspections as well as audits and trainings or reviews and sponsor and internal CQA audits, the team worked in staggered shifts, and always there was somebody or the other present from the team to make sure that in-life inspections were conducted, study-based inspections were conducted, and that we were able to host audits, and all the interactions that we had over video calls and Zoom were recorded. Our training program was switched to online mode, and the QA really worked very hard to make sure that all our SOPs, our methods, our guideline trainings were all also shift to the learning management tool online, without really compromising on data review as well as facility and assay review. The other key aspect of large molecule work is critical reagents, right? Our critical reagents can really make or break our assays. So the critical reagents, as you heard Dr. Darshana Jani speak already, could be anti-EADs, they could just be proteins, they could be targets, they have to be labeled, they have to be sourced, they have to be, go through a biophysical characterization. So the first and foremost was about procurement. We wanted to make sure that the pandemic interruption really does not cause an interruption in our procurement and logistics, because if that interrupts, then anything that we do to work during lockdown will not make sense if we don't have critical reagents and material. Therefore, we really dedicated a pandemic management team which met every day, and sometimes every day, and mostly every other day, to review situations and really continuously evolve with the evolving situation, and changed the way that they really made procurement, changed the way that logistics was handled, and about how we paid invoices, and that last but not the least, how were we actually managing our inventory. So all this, we were lucky that we started thinking this well in advance in February 2020 itself. Therefore, that really helped us not only during the first wave, but also really cruise decently during the second wave as well. And at the end of it, the results really that we, when we came back and we checked and we saw that the results were indeed pleasant. We found that, we were able to maintain our focus on speed, on the quality as well as compliant. And 99% of the times, we were proud that we were able to run on time, make on time deliveries of either plans, reports, data, database logs, etc. And the and another pleasant part was since we were forced to really reduce paper reviews, we had about 70% of paper reviews was reduced. And this is something that all of us have been wanting to do, to really convert our lab completely electronic, data friendly, but somewhere we still call ourselves hybrid. But the pandemic really pushed us to move away from paper and to really become much more electronic than we thought we will in a matter of few months. QAO inspections were conducted as a large number, 190 inspections, which meant these were audits, these were in-life inspections, these were database log inspections, anything and everything to do with data as well as studies. And at last but not the least, our employees also reported that about 30% of their daily time was actually free to focus on their personal lives, because we were not traveling up and down to work, and we were really able to optimize our time when we came to work on certain days and when we were working from home on other days. So this also really brought a smile in a way to our employees. So by making small but even forced changes in human interactions, really, our group was able to undertake all the required large molecule bioanalytical studies that is required as per GLP, GCLP compliances. So all the way from assignment of a study to the SI, to the SDPI, setting up the study, to samples that are being received into the laboratory, that they are accession, and then the work lists are set up. The sample accession team gives the samples to the PI or the analyst, and then analysts run their studies. They give the data to the PI goes into the QA. First after QC, the PI goes into the QA with the data, and then corrections and the clarifications are made, and finally the data or the report is submitted to sponsor. So everything that we had to do in terms of our workflow was actually done in spite of a change or a cease or pause in the way that we interacted with each other physically. So we still continue. So regardless of the pandemic, regardless of the changes that we were forced to make, we really still continued, and we are still continuing running interesting targets, interesting assays, and studying interesting targets to set up fit for purpose PK, immunogenicity, and PD assays. I hope I'm able to in the next meeting or so describe a little more technical and scientific aspects of the assays that we do, that you see here on the screen. So everything went well. It was not easy, but it did happen, the way that we did not anticipate it will happen. But at the end of the day, we were really cruising pretty well in terms of supporting all our clients, our patients, as well as regulators. Now, the added, I could call it a pressure, but at the end of the day, it all worked out well, was that during the pandemic around March 2020, our team, used the large molecule bioanalytical skills that we had developed, the fit for purpose assays, the fit for purpose validation expertise that we had, to really support COVID-19 assays. We set up one of the earliest COVID-19 testing laboratories, and with our experience with GLP certifications and GLP inspections, we were able to get a certification from NABL and ICMR really early. And then we moved as soon as the laboratory was set up. We had staff working, and we had almost 80% of the work we do today is for the government of Karnataka and government of India, and in the COVID testing RT-PCR laboratory. And besides that, having our own laboratory also gave us the leverage, the freedom to test our own employees. So we ran our routine testing. We made sure that we were able to track, isolate, and treat. So when the RT-PCR lab was settled and it was running well, we then focused our attention again to developing assays for, against serology assays for SARS-CoV.
Hello.
We lost
Hello.
her, I think.
Yeah, I think her connection. Yeah, she's back again. Yeah.
Hi, are you able to hear me now?
Yeah.
Yes, yes.
Okay.
Please proceed. Yeah.
Yes, I'm sorry. So then we moved our attention to developing SARS-CoV-2 antibody assays. We developed IgG-S1 serology assays. We did surrogate neutralizing antibody assays. And when we validated these assays, so somewhere or the other, the bioanalytical skill that we had developed over a decade and a half really, really helped us in not only supporting the community, but also in doing it at a pace that we had not anticipated we would ever be able to do. So that was really something that we are happy and proud of. So I think, I don't know why my screen is not moving. Okay, so I guess that is really the end of my presentation. So this is really how we managed bioanalysis during the COVID-19 pandemic time. And this is really how we worked, and we moved and transitioned to different ways of working. And we were still able to support the studies and as well as the patients and therapeutics that we had contracted to support. So thank you very much for your attention. I'm happy to take any questions or wait till the end of all the talks and then meet with the panel.
I think we're going to have a question and answer session, Aparna. So
Okay.
there is a question on the chat, like one of the biggest challenge of COVID-19 is getting the reagents, kits, and chemicals from global vendors. How you guys overcame that issue? Like how you ordered those stuff in advance so that the sponsors' requirements are met on time?
Yeah, so you said it, Pat. So all we did was we did this all in advance. So before our borders closed, as soon as we were following global news on an hourly basis, if I may, and before the borders shut and before there was a disruption in the in the shipment service, we quickly we worked in advance and we figured out what is it that we need for the next 6 months. At that time, we did not know about the second wave or anticipated, but what would the things that we would need for the next 6 months. We just placed an early order. We spoke to all our sponsors. We told them what is it that we will need and what is it that we have, and we just planned well. So that was something that really worked out well. However, the Indian government also allowed cargo planes to land, which meant that our material, our shipments could definitely land. There was some staggered clearing at customs, but clearing did happen. So against a 5 to 7 international travel time, I believe we faced a delay of about maybe a week or so, but our reagents did land safely.
Good to see that 70% paper not being used, saving the trees, and also
Yes.
employees working from home. You might have saved some pollution too with the vehicular traffic. So other question I have is, I kind of lost my thought there. Yeah, Shafali, you have any questions before I can come back again? Shafali?
I just want to thank Aparna for this lovely talk, and and being a real Corona warrior. Each one of us faced the pandemic and had their with their own challenges, and it is so nice to see your team managing so well at all the fronts. And it was a difficult time. I don't feel. So you already had the RT-PCR set up and the SOPs in place, Aparna, before the pandemic started, right?
Oh no, we had to develop everything from scratch. The COVID-19 RT-PCR assays we developed, from scratch. We set up SOPs. We looked for facilities where we could run, where we had, hoods with the viral burnout. So, and we assembled a team across discovery, development, bioanalytics, a team of scientists that worked to develop these assays, to set them up, to get them certified. So everything just started from scratch.
I think there is a YouTube video of that whole
Yes.
venture, I think, and it's also on the LinkedIn too. If people want to Google that, it's also there on the LinkedIn, I think. And the other question as I have is like, how much production loss was there during this period? Was there any productivity loss because of business loss during this period?
No, there wasn't. In terms of people productivity, we did not see a loss because we had people either working from home or working from work. And, touch wood, we also did not see a business loss. There was a little bit of a slowdown because some of our clients, did not have studies running as fast as they had imagined, especially when it comes to patients. So there was a little bit of a slowdown in terms of receiving samples, but it all got made up by the end of the year.
Yeah, one follow-up question is that people were worried about COVID positive samples being received at labs. So what was the action plan taken by you to handle those, not knowing those samples were COVID positive or not?
That's a very good question. And here, the one thing that I want to do is to thank all our clients and our sponsors that were so vigilant and that were working with their sites. So sites really determined if there were COVID samples, and if there were COVID samples in the manifest, those samples were removed in the beginning and then they were sent to us. So till we got our COVID protocols all done, we were really not handling COVID samples. When we got our COVID serology protocols done, we were then able to handle COVID samples with adequate biosafety.
I remember
but
you
I
asking
don't think, yes.
I remember asking whether heat treatment can be allowed or not, and that
Yes.
was a big
Yes.
no-no for the
It was a
samples.
conversation we had, and we had a similar conversation. But till date, I have not had a client, come in, or a customer, or a manufacturer coming and tell me, there's a COVID-19 sample that we would like to import into India. I believe it's already blocked at sites.
Okay, gotcha. There is a question on the chat. What all parameters considered to be fit for purpose validation? This is for fit for purpose validation.
Yeah.
Question from Avinash. Yeah.
Right. So I'm not sure this is in what context, but I think I will talk about it in the PK and the ADA context. So when you look at a fit for purpose validation, in a PK, for example, while you're looking at accuracy and precision, you're looking at dilution linearity, you're looking at stability. The two most important aspects of the validation are selectivity and specificity. That really, in a way, makes it fit. It makes it fit because you're looking at target interference, you're looking at matrix interference, you're looking at any other kind of, rogue components of proteins in the matrix that may come and interfere. So, fit for purpose validation, again, as I said, accuracy, precision, dilution, linearity, selectivity, specificity, sensitivity, as well as stability in a PK assay. When it comes to an ADA assay, a fit for purpose validation will first involve the assay cut point. We need to be sure that we're marking the assay cut point very clearly and very methodically with adequate reasoning and statistical tools. And then we move on to judge precision. Again, the fit for purpose comes again, from the selectivity, the specificity, and then of course the sensitivity of the assay. And sometimes we do run stability for long-term stability for ADAs, or sometimes, or most times we don't. But we do a freeze thaw, a bench top, and a short-term stability for all our validations, be it PK or ADA or immunogenicity. And the same rule is followed when we're working with cell-based assays, where the fit for purpose is a little more stringent in a cell-based assay because you're looking at various different cellular cascades and cellular components that may probably be interfering in your functional format. So these are the main aspects of fit for purpose validation.
Okay, I think in the interest of time, thank you very much. It was very informative and enlightening, the trials and tribulations of facing the unprecedented situation and handling that throughout this 2-year period. Congratulations, Aparna. Much appreciated.
Thank
Okay,
you.
I now like to introduce my colleague, esteemed colleague, Amarnath Jaiswal, to introduce the next speaker. So, Amarnath, it's all yours now.
Yeah, I
Thanks,
I think
Pat.
it would be a nice idea to just give this
Oh, yeah.
certificate of appreciation to
Sure.
Aparna before
Okay.
Amarnath could introduce
Yeah.
the speaker.
Thank you.
And,
So my
and,
apologies.
and before introduction to Arjun, I had one question to Aparna.
Oh, okay.
I mean, we we all know that that the last year, even in the present situation, also the COVID pandemic was the new experience for everyone and across the organizations. And as you said that, I mean, the majority of companies they started working on site, the offline working from home. And there was a lot of the question related to the data integrity and the signing of the documents. I mean, the people had a
Yes.
practice to do the paper based signature. And when we suddenly change our thought processes, when the people are not allowed to come at the offices, particularly those who are in the admin job or the QA functions or the project manager kind of thing. So how did you cope up those situations to make sure that that the work is not being affected and my data integrity is still considered to be the valid and reliable? I mean, what are the changes you have made quickly in terms of the SOP procedural and all?
That's a very good question, and I think I missed covering it in my talk. So we switched to, for our signatures, we switched to DocuSign and then to Adobe for electronic signatures. We procured many licenses to make sure that each of us have license to go and put that signature. And of course, there was always the email approval. If there's an audit response report or something that we had to approve, there was always an email approval, and the emails were all printed or scanned and kept in the study folder for traceability. So that way we made sure that anything that we did, we had adequate traceability to what we were doing, and the date time stamp was always present, and audit trails on all the equipment was always there to really understand what is happening on the bench.
Great. So that I believe that has been specified in the SOPs,
Yes.
right?
Yes, and the SOPs of course specified it. And when we could not really immediately go and revise an SOP, we made note to files, and we made management documents to inform the staff as well as a regulator that will come to audit to say this was done with a plan, and this is a procedure that we will follow.
Thanks, Aparna.
Thank you.
Okay, so
Thank you.
Yeah, thank you, Aparna. As
Thank you.
Shefali mentioned, would like to present the certificate of appreciation. Thanks for a very enlightening talk about how large molecule analysis was handled in the pandemic COVID period. And I'm sure there'll be a few more questions. So would really appreciate if you can hang around till the panel discussion, and we'll take up other questions during that time.
Certainly.
So thank you, Aparna.
Thank you very much for inviting me. Thank you.
Thank you.
okay.
Yes, and Patanjali, would like to thank you also for wonderfully moderating this session. I know it's a little, not little, I think half an hour over midnight, and it must be really late for you. So thank you. We really once again thank you for your active participation. Thanks to Shefali as well. And now I would invite over to you, Amarnath, our moderator, for the next two talks along with Srinivas.
Have a good webinar, guys. Bye.
Thanks, Patanjali. Thanks. Okay, so let me introduce our next speaker, Dr. Arjun, who has a experience in the field of the bioanalysis, and currently is the owner of large molecule bioanalysis advice. And as I said, that is the more than a decade's experience in terms of the bioanalysis. He was a steering committee members and the core member at the European Bioanalysis Forum as well. And he was involved in the many drugs and the vaccine development program up to a phase 3, starting from the development phase to the successful health authority submission. During this preclinical clinical study, he was also responsible for the development and validation of the PK immunogenicity and the biomarker assay and the subsequent sample analysis. Currently, he is working as a bioanalytical consultant for the several pharma companies and the monitors the bioanalytical work performed for the companies at the different CROs. Today he's going to talk basically on the PK assay development, and the most importantly, I mean, everybody's trying to look at the reliability of the data, and one of the key aspects is that the reagent. So Dr. Arjun is going to talk about, you know, the selection of the right reagent for the assay development, and then the subsequent validity of the assay through a validation and until the report submission. So I would invite Dr. Arjun, and then he can probably give a, you know, his talk, and I hope that the participant would really get some benefit from his talk. Over to you, Dr. Arjun.
Thank you for the introduction. Can you hear me?
Yes, absolutely, we can.
Okay, let me share my screen then. Okay, 1 second. Yeah. Okay, good morning, everyone. So the presentation of today I'm going to give is about PK assay development and validation. It's entitled PK assay development and beyond, which means that it's actually a kind of oversight of the whole procedure. 1 second, I need to switch something off. Once, okay. Because this, I'm going to do it automatically. And it's quite difficult. No. Okay, good. Then first, an overview of my talk. First, I will start about PK assay setup, then assay optimization, followed by pre-validation as soon as we have that assay in place, and then the strategy of validation, followed by the actual validation itself, reporting phase, and finally the assay lifecycle management. But first, to introduce myself, my name is Ian Companion. I'm owner indeed of Large Molecule Bioanalysis Advice, and LMBA for short. I have more than 15 years experience in bioanalysis, and I'm a bioanalysis consultant. So I work for different pharma companies, and for them, I'm a kind of intermediate between the pharma companies and CROs. And as such, I'm a guide, monitor of development and validation of PK, immunogenicity, and biomarker assays. And I'm also involved in the reporting of data and also the subsequent submission to regulatory agencies. So, okay, so what are we actually aiming for when we want to develop and validate a PK assay? Well, we have to develop a quantitative assay for drug level determination in tissue matrix in a precise and accurate way. That's what we do. So as a scientist, you should know you are responsible to set up this assay and to defend the data to support that clinical program. And regulators provide guidelines to aid you during that development and the validation. In fact, these guidelines are more or less a roadmap for you to use to, you know, get your assay in place. So your assay starts with good critical reagents. So that is like the basis of your assay. And what should those good reagents then do? They should be specific, they should be selective, and they should result in a sensitive assay. And basically, that's what you want to see. So you have to invest in development of good reagents. If you do not have good reagents, well, you basically will not have a good assay. And that gives you a lot of problems later during the clinical program. For example, use anti-idiotype antibodies in case of monoclonal antibody therapeutics. I would advise to, you know, get away from commercial produced antibodies, general antibodies. They may not be very specific, and also the production may stop in a certain point in time. You do not have control over these assay reagents. It's also important to have control. So which assay formats do we actually use? Well, we have, of course, antigen capture, in which the antigen is coded to the plate and detected by the drug, which is made for the target. And then the drug is, detected by a general antibody. This is the format I usually see, a bridging assay in which you use anti-idiotypes against your drug, which is a very clean assay, very high acceptance rate. Of course, you also have an combination of that, like an anti-idiotype capture, which captures your drug, and then detection by a general antibody against the drug. And of course, you have also kind of intermediate format in which you use an anti-idiotype, recognizing your drug, and then the target is the tracer of your assay. So these are basically 4 different formats. Then you need to know you have a free and a total assay. In a free assay, you have your anti-idiotype, and this is all for antibody drugs, which recognizes your drug, but then blocks the targets, the place where the target binds. So this is actually a free assay. So you only detect the drug. And a total assay, which recognizes the drug, but is not blocked by the target. So you will detect free and total, and drug in context of the target. And you have also an assay which recognizes the complex of the drug and the target. So as I said, this is all monoclonal antibodies, therapeutics, but you can also have these kind of formats for other proteins. Okay, different platforms. So which platform these days will you use? We have the ELISA for decades that still is used sometimes, but now we also have a GyroLab, which has a very large dynamic range, can have a low sample volume, very fast in development. LCMS, in this case, you have a direct measurement, so you don't have, usually I don't have like capture detection antibodies, but this is only total assay only, because you break down your drug and you will detect only drug, not in context with your target. MSD is more or less the standard platform. It has a larger dynamic range, and the new kid on the block is the Simoa, which is very sensitive and has also a very fast development. So this is actually quite new, but also already used for PK assays, but very sensitive assays. So now we know, well, now we have the reagents in place, and we know which platform we are going to use. Then the optimization of the assay comes. So that is one of the most important stages. That's the backbone of your assay, together with your critical reagents. How would you do that? You can do one factor at the time of that. Well, most of the CROs do this. They have a checker box optimization, capture and detection concentration are varied, and you just select the best one. And you go by step by step. Another approach is the design of experiment. This approach, you can vary different factors at the same time. For this, you actually need a software, for example, JUMP. And the software on the left side, you can see you can import all your factors, and then the program calculates the different assay runs, and the outcome are a lot of factors which can be combined. And from these combinations, this program calculates what is the optimum. So this is a really very fast way of optimizing your assay, but the assays become very complex, but it goes fast and you have a very good assay in the end. Okay, one of the most important, besides all the other things, a very important part is specificity of the assay. The assay has to be fit for purpose. Our previous speaker talked about it as well. So interference, in fact, also shows already a little bit about the specificity. The drug will, recognize specifically, if all is well, the target. So if the target interferes, the assay should be kind of specific. But also, you should be consider to use an interference by irrelevant antibody. So an antibody which looks like your drug antibody, but is directed against a irrelevant antigen. In the case of monoclonal antibody therapeutics, this will work, and this should not interfere in the assay, right? Matrix effect, one of the things you should determine quite early in development is to see whether the matrix effect has an effect in your assay. So this is the time you would like to see the minimal required dilution, In this case, you compare different matrix levels with assay buffer only, and if the curves are, on top of each other, that is your MRD. And usually that MRD is between 1 to 10, 1 to 100, depends. And it's also good to already to test different individual matrices. This you do with your pooled matrix, but it's also good to see what different individual matrices already do, so you know what the whole population will, what the effect of the whole population will be in your assay. Hook effect. Another thing we need to test quite early is hook effect. This is the phenomenon where you have an increase of your analyte, or in this case your drug, and at very high levels the signal starts to drop. And this is what you do not want to see in your assay, because this can lead to underestimating of your sample, your drug concentration in your sample. So that should be tested already very early on to see whether your critical reagents are actually work in this assay. Then the determination of your assay range. I know many of you are from CROs, and maybe the sponsor will already tell you, okay, this is the range we need to measure in. So they give you more or less an assay range, but you should determine the LLOQ and the ULOQ. But sometimes that LLOQ is just given, like, okay, 250 nanogram per milliliter is enough, and they call that then the LLOQ, but the real LLOQ may be even lower in such an assay. And at that time you will determine the correct curve equation, usually so that mathematical model to plot the curve, usually 4PL, 5PL with different weighings. I also saw other mathematical models applied, but usually this is the what is used in such assays. Okay, so we now are at the stage that we almost have a kind of assay in place. So at this time, calibrators, which actually are very important, as you would understand, need to be set because they determine the curve. Usually people take at least 7 levels in the assay range and add some anchor points. I would advise to use more than 6, so you can delete a few outliers or whatever happens, but you need to have 6 in your assay range by, that's also in the guidelines. Usually those calibrators are made fresh before every run, but frozen stability can be determined as well. And that is, if you have that, you can also freeze your calibrators and use them for that can be handy. Quality controls, don't forget, quality controls should mimic more or less your clinical sample. You should have 3 times LLOQ at the inflection point, and 80% of the ULOQ is mostly advised. I would advise to avoid using QCs at the same level as the calibrators. Some CROs just ignore this, but well, this is something you should consider. And the storage should be similar to the clinical samples, and you should produce enough QCs to cover stability experiments, including long-term stability. And long-term stability, in this case, the QC is really your clinical sample, because that will determine whether you can store your sample for a certain amount of time. Okay, working stocks. Please make working stocks from the drug you will receive from the CMC department or from your sponsor. So that means that you spike your drug in blank matrix at a level easy to use for your assay. I've seen that sometimes not happening, but I would advise to do that. And preferably you should use the same drug lot as used in the study. 2 working stocks should be made, one for calibrators and one for QCs. The reason is that you avoid mistakes. So if you make a mistake in dilution in the calibrator part, you will see that in your assay. But if you make a mistake in one working stock, then the calibrators and your QCs are the same. So per guideline, this has to be done only once, but I would advise to always do this. And also determine the long-term stability of these working stocks. It should also be monitored. Okay, so now we are in the stage that we are pretty much confident that our assay is fit for purpose and that it can, you do what it should do. So in this final assay method, at this stage, the final item method is issued, and all test items, all items which are relevant and already included in the validation are now tested just before the validation to get more confidence. Important, of course, is precision and accuracy. You can do, for example, 3 or 4 assay runs on different days by 2 analysts to get some idea about the precision and how accurate your assay is. Maybe this is also the time to start to do some robustness experiments. Robustness, that is small but deliberate changes in the test conditions. So different incubation times, for example, that will give you more flexibility during sample analysis. Because you know that your assay can be incubated for a longer time or shorter time. So if there are mistakes, then you don't need to repeat your assay immediately. So and all results are written in criteria, then your assay is ready for validation. So different agency produced guidelines on bioanalytical method validation. One is, of course, the FDA guideline from update in 2018. EMA since 2012, they already had that a guideline, which is to which this guideline of the FDA is more or less modeled. But now we have a third one from the ICH, which still is draft in 2019. That's the bioanalytical method validation M10. And now people already use this as well, but most of these guidelines are, most parts are similar. So what is actually the definition of validation, which is the degree of validity of your measuring device? That's what it means. So what it doesn't mean is, does the assay, can the assay do what we say it can do? So for validation, parts of it that they can fail. That is you have you set criteria, and the process is that you first draft a validation plan in which you indicate the criteria and the statistical analysis, what you would do a priori. Then upon signature of your study directors, you can start experimenting. Then you have your experimental phase. After this, the data analysis and everybody happy on the data, then we actually have a sample, an assay in place for which we can use for sample analysis. And after this, the reporting phase will come, which is quite an important phase, by the way. Well, the items we need to test during validation are precision, accuracy, dilution, linearity, hook effect, stability, target interference, selectivity, and of course you can add more if you like, but this is like, the most important. Precision accuracy, that's more or less the core of your assay. Of your validation, you need to determine 5 levels of analytes at the lower limit, upper limit, and 3 in between. These actually are your LQ, MQC, and HQC. You analyze them at the start, middle, and the end of the run. Have to do 6 experiments at least over different days by at least 2 analysts. And you determine your intra and inter-assay precision and accuracy by ANOVA. We should not forget the total error, of course, and the criteria are indicated in the guidelines. Within 20% CV and RE, or 25% at the LQ and ULQ level, and the total error, which is the sum of the CV and the RE, should be within 30% and 40% at the LQ and ULQ level. And you also should report the PNA of the other runs, but I would advise to do that separate to keep this as an isolated experiment. Dilution linearity, which is in fact the test which tests how high what your maximum dilution should be above the MRD. So in samples which have a concentration higher than the ULQ, the question is, can we dilute that in the curve? That's what you prove with this experiment. So you spike your analyte in blank matrix and then further dilute it in matrix to mimic a high level sample. And the way you dilute, that's for example, if you pipette 10 microliter to 90 microliter and then do another dilution, that is validated as well. So you have to, in fact, you better stick to that dilution scheme. This is also a part where you can test dilution QCs. I didn't mention that before, but you can take those along during the precision accuracy. And those dilution QCs are just the QCs for the high level samples. And you can take them along during sample analysis when you have to do reruns or with dilutions higher than your MRD. Hook effect, already discussed. Prozone, that's also another name. That should be tested as well. And you assess that by spiking a very high level of your analyte, and that signal should be equal or higher than the ULOQ. Should not be lower, because then you're in this zone. Stability. Well, stability is more or less the processing conditions which are used during the whole procedure after isolating a sample from a patient up to processing and assessing it in a lab. All these steps should be understood. The stability should be tested. So that is usually storage stability in the fridge or the freezer, at the bench top, freeze-thaw stability, and also long-term stability. And that can actually last up to 2 years, depending how long that study takes. That is done, by the way, at the same condition as used at the clinical sites. So you should make enough QCs to determine that long-term stability. And you can also consider to test other stability conditions like full blood stability, for example. Target interference. If the target is in fact present in the matrix, then you should determine that interference at different target levels, in which you incubate your target with the analyte. And in the case there is an effect of your target, then you know that it is a free assay and not a total assay. Selectivity, quite important, already tested during the development a bit. That is the effect of the substances in the matrix on the assay signal. Should be performed in at least 10 individual matrices for every species or disease indications. And also to be performed in hemolytic and lipemic matrix, which occurs, a lot. When you do studies, you always see these samples coming by. But they can also be performed in rare matrix, like ecteric matrix, for example, depending on the disease state. And the conditions are, spiked and unspiked, LQC or HQC levels. And the criteria is that 80% of the matrix should pass the preset criteria. And in the unspiked matrix, the signal should be below the limit of quantitation. Parallelism. So now we're actually done with your validation, but there are other parts of the validation should be tested during sample analysis. And one is parallelism. That is to assess the linearity of the actual study samples containing a high level of analyte. Don't forget, your QCs are samples in which you spiked your analyte yourself. In this case, you deal with samples from patients which have been injected with the drug or otherwise, and that should be tested. You have to select Cmax samples and dilute them into the range. So you should have like 3 levels in the assay range would be best. And you just check whether that comes back, if you calculate back, if that is comparative with your comparator level. Incurred sample reanalysis is something you need to do as well. And that is just by reanalyzing incurred samples and to confirm that accuracy in those samples, 10% of the first 1,000 samples and 5% of above. And the minimum number usually is 20 to 24 samples. The criteria is that 2/3 of the samples should be within 30% of the original results. And these both items, they can be amended to the validation report, which is handy because then you have it in one, in one report. But you can also report it in the sample analysis report. Okay, then reports. Yeah, of course, reports should be very clear and concise. Top line tables, that is something which makes it much clearer for the regulators when they have to review such a report. And in fact, FDA issued a guideline for tables for inclusion in the M271 document, which is the document which is also submitted to FDA. And this is the guideline. This is an example, part of the table which they show, in which they ask a lot of items to fill in, method description, assay range, what have you, and of course the observed, CV and accuracies. So I made a table to put in a report, and here it is, which also indicates the analyte, the technique, and also everything about the LOQ, the ULOQ, and everything. And this works pretty good. If you put this in a validation report, that is so handy in the end when you do the submission, where you don't have one report, but many reports. So people, the sponsor can then just copy paste that in their that table. That saves a lot of time and also avoids mistakes in the large submission document. Then finally, assay lifecycle. Critical management system that should be really well in place. Because your sponsor, they don't want to hear 2 days in front, okay, we need more reagent because the expiry date is almost there. And they also don't want to hear, okay, we need more reagents, and then a couple of days later, oh, we found a box in our freezer. That is not good. You should have that management system really working very well. And also reagents made to be bridged anyway. But there is a paper on how to deal with critical reagents written by Sutan Pill of EBF, which is worthwhile to read, by the way. Then trending. Trending is something more and more sponsors want this. It should be monitored just in time, your assay. It should start during the validation, and that should be performed during the whole life cycle assay. And that is something, as a CRO, you should have this actually in place. You should have a nice trending system so that you can also share those data with your sponsor, which can see that how your assay is performing. Quite important. Okay, so this is actually the end of my talk. Thanks for the attention, and I wish you good luck with your assays.
Thanks, Arjun. And I think you have again refreshed all the guidelines, the validation parameter. And it's always good to have the people have the refreshment on all the validation parameters. So I didn't see any question, but I have 2 questions for you. One of the question is related to the stock preparation. So if you look at the FDA guidelines, the FDA state that the stock should not be prepared from the reference standard, which is near to the expiry. So I just wanted to know your thought process to understand what do you mean by the nearby. It means we can consider for a week, a month, a couple of days. I mean, it's always a debatable question.
Yeah, well, a good question. What is that then? Yeah, for monoclonal antibody drugs, they are of course stable, but yeah, the expiry date is the expiry date. So you just need a retest date from your sponsor. If that is not there, you have to bridge to a new drug lot. That's what you should do. But when there is an expiry date, if you have your expiry date on the label of your drug and you have to make stock solutions for that, that's what you mean, right?
Yes, absolutely. And let's say the expiry date is tomorrow, 10th of October. So can someone use on 9th or 8th or 7th? I mean, it's always a question mark.
Yeah, I know CROs who do this. So they have their drug, which is almost expired, and then they're going to make QCs very fast, like because tomorrow is the expiry date, and then they put a new expiry date on it. Yeah, that's not really fair, is it? I mean, but that's not really, actually, I think it's a bit strange. I think that if you make stocks of your drug, and your drug has a specific expiry date, that is the expiry date, which it is. So if that is expired, you cannot use your QCs anymore.
Absolutely, nobody can use. I mean,
no. But one thing, there is still a problem. what about your long-term stability samples? You made them way in the beginning. For this, you can say, well, then the drug is expired, so we cannot use the LTS samples anymore. But no one does that. They just keep on using that. But that's fair, because when you inject the drug in someone and it expires, so you take the samples, then that same drug is in that sample. And if that drug is expiring over time, you have the same situation. So your QCs for long-term stability, they mimic your clinical samples. So for this kind of samples, it doesn't really, that's not applicable. For your long-term stability samples, I would say it's not applicable, but for all your other samples like calibrators or QCs, in fact, you should, consider that it expired dates. That's my opinion. I know that a lot of CROs do it differently.
Absolutely.
Yeah, but
I mean,
for monoclonal antibodies, for drugs, they're usually very stable. And if your long-term stability indicates that your sample is stable, well, okay.
Yeah, I mean, the best way is to, because as you said that the different CRO has the different practices, and as long as it is specified in the written documents that the near to expiry would be considered for a week or 2 days, 3 days, I think as long as it is specified, the agency would not have any problem.
Yeah, you know what? Documentation is everything. That's very important. You should document everything very carefully. And if you have that in your SOPs, then it's fine. But regarding the drug, it depends on your drug. Maybe there are drugs which are really unstable. That's a different question. But
absolutely.
Yeah.
So there is one more questions. I mean, if you look at the different CROs and different industry have their different practices, and when it comes for the plasma stability, right? The people used to take the blank plasma, blank serum to spike their drug product to evaluate the biological samples, the stability. And we have seen that, the many of the times people used to take the blank plasma or blank serum, which has undergone a multiple cycles. So as you are aware that when the blank samples undergo a multiple cycles, that has also a tendency to protein get denatured. So do you feel that the organization should also design an experiment to make sure that when they are using a certain, the blank plasma or a blank serum for their actual validation experiment, do they also need to establish that even their blank sample has undergone a multiple cycle, still it is giving the reliable result? I mean, the protein is still not completely denatured, and still the analyte has the bond with the plasma or serum protein. I mean, we used to do it in our lab, but I have not seen many of the CRO that they don't have the practices to do it.
I never seen this.
Yeah,
but then again, most of those projects are antibody projects. Nobody bothers. They of course they test the blank serum in the beginning, so that for example for immunogenicity assays that there is no pre-existing immunity in there. That is true, but I've never heard of. But it can it can be that depends on the drug. So most of those drugs are antibodies. Everybody knows that doesn't have an effect on the drug. That is not an issue, but there might be other drugs which interact with proteins in the sample, which can have an effect. Yeah, that's true. That depends on the drug, I would say. But for all the projects I'm working with now, never saw this before.
Yeah, because that was the raised by one of the auditors, and several years ago with one of the study audit, and they have realized that the plasma which is used for the method validation has undergone almost 7-8 cycles. Though the nature of the analyte was not closely bound with the protein, but that was the point raised that there may be a possibility if your analyte is intended to be bind with the protein, that you are not really demonstrating the actual validation. So that's the reason. I mean, we have not seen many of the CRO, but there would be in the rare cases.
Yeah, it's interesting that you say this, because it never occurred to me, but it can happen. Yeah, I think that has something to do with the drug. Yeah, there are also many things in such guidelines which we think, well, is this necessary? But all these regulators, they see many studies. So those things in the guideline, they just don't fall out of the sky. There's a reason that they put it in, but it depends on the drug. And most of the antibody drugs, they are not like bothered by that.
Correct. Thanks, Arjun, for giving such a wonderful talk, and I think the people will refresh their minds, on the validations experiment.
Thank you
And
very
before
much.
I invite Srinivas for the next speaker introduction, I would like to just present this a token of appreciation. And thanks, Arjun, and see you in the next the next year if it is in persons.
Okay, thank you very much.
Thank you very much. And
Good
I
luck.
would like to invite Srinivas to give an introduction to the next speaker.
Yeah, and Arjun, if you are available after this talk, then we would really be more than happy to have you as one of the panelists for the panel discussion immediately after this talk. So if you are there, then that would be great. Yeah.
Great. Yeah, I will.
Thank you. Thank you.
Thank you. Over to you, Srini.
Okay, that's great. Thanks, Amarnath. It was wonderful talk of Arjun's. Now we're coming to the next session, next talk in this monthly theme of biotherapeutics development. So next we have Jayan Albert from SciEx India. Jayan is a sales development manager at SciEx with a high level of scientific understanding and customer needs in the field of analytical instruments. So we are bringing someone from the tech side now. Jayan has about 10 years of rich experience in analytical field and is responsible for achieving short and long-term business objectives of the company. Of course, he's working for a specific pharma and CRO vertical. Earlier, he was also associated as application scientist, supporting to customers in method development, software training, troubleshooting, as well as maintenance of different range of SciEx instruments, especially mass specs. He's also a SME in developing small and large molecule quantification methods in various biological matrices using LC-MS technology. He also worked as research scientist with Sun Pharma in regulated bioanalysis, and he holds the postgraduate degree in pharmaceutical sciences from BITS, Poland. Jayan, welcome to you on this platform, and over to you. I think we are looking forward for your talk on biotherapeutic peptides, quantitative challenges, and SciEx solutions. Over to you, Jayan.
Thanks, Srinivas, for the wonderful introduction. Let me share my screen. So can you confirm, Srinivas, is my screen visible?
Yes.
Okay, perfect.
And you're audible.
Yeah, thanks. So I welcome everyone who has joined today for this presentation on biotherapeutic peptides, the quantitative challenges and SciEx solutions. And this is my agenda for today's presentation. We'll be starting with the overview of the therapeutic peptides and the number of therapeutic drugs or the therapeutic peptides under development, how many of them are approved, and the challenges involved while quantifying in the biological matrix using LC-MS, and SciEx being a leader in this industry, what SciEx solutions to address and overcome all these challenges. We'll also talk about some interesting data we have developed using the SciEx LC-MS systems in this presentation as well. The therapeutic peptides overview. And peptides are the linear polymers of amino acids linked by the peptide bonds. That is the covalent bond formed between your carboxyl group and the amino group. And use of them for therapeutics has started with insulin in the 1920s, way back in 1920s. But in the last decade, the interest in the therapeutic peptides has increased tremendously. The three simple reasons for that: they are very highly selective, they are efficacious, and relatively safe and well tolerated compared to other small molecules. Right, but the use of these naturally occurring peptides for therapeutics is limited by the poor pharmacokinetics, like short half-life and the negligible oral bioavailability. But the emerging medicinal chemistry techniques helps in designing of the novel peptides with extended half-life and making them really resistant to the proteolysis to improve its pharmacokinetics and oral bioavailability. And if you look at the literature today, more or less 70 peptides have been approved in the United States, Europe, Japan, and more than 150 are in the active clinical development program. So it's important that the BABE or DMPK labs should develop their expertise to tackle these future requirements. And traditionally, these proteins and peptides have been quantified using ELISA methods. However, it has its own advantages and disadvantages as well. Looking at the major disadvantages of the ELISA or the ligand binding assays, they have the limited selectivity or specificity compared to LC-MS/MS analysis. And also, it has its own inability to differentiate its metabolite or analytes, metabolite or the analogs of the drugs. And also, there is a need for antibody before performing this kind of analysis. So definitely LC-MS/MS has advantage over the ligand binding assays. And we see a shift in the platform of labs moving from the ligand binding assay to LC-MS/MS, considering its advantages. Although LC-MS/MS is the technique of choice, but it is not without challenges when used for quantification of proteins and peptides. The reason is the chromatographic and MS behavior of these proteins and peptides is very different from that of small molecules. So what challenges we may face? What challenges your lab may face when you're moving from the small molecule analysis to these peptide quants? The first one is the low level of quantification. As these therapeutic peptides are being very potent, the low level alloQ in matrix is a primary requirement. However, it is limited by its physiochemical nature, forming multi-charged ions in the ESA source, and also the poor fragmentation in the collision cell of the MS. As we all are aware, in triple quads, the sensitivity is based on the intensity of your Q3 fragment. And when peptides are known for poor fragmentation, especially the cyclic peptides, it further aggravates the quantitation challenges. The second one is the matrix interference. The matrix interference with the analyte of interest tend to happen as it involves measuring a small peptide out of multitude of proteins in the matrix. And sometimes this may lead to matrix effect or even contamination of your ion optics as well. The third major challenge would be your recovery. The recovery could be an issue, especially at lower level due to the stability of your peptides or sometimes even non-specific binding issue. The fourth major issue could be the adsorption issues or the non-specific binding. The adsorption issues are very predominant. The proteins or the peptides, it may stick your pipette tips, your polypropylene tubes, your SP cartridges, vials, autosampler components, etc., etc. And how to mitigate that? So in general, a suitable concentration of a bovine serum albumin are added in your sample to minimize this non-specific binding. And we generally sometimes use the low binding tubes as well. These are also available in the market while processing these peptide samples. And many times we see that conditioning the chromatographic system with crash plasma would also help in minimizing this adsorption issues. And as I discussed before, due to this adsorption issues, the auto sampler carryover and column carryover is also very, very usual challenge we face in the lab while developing the methods for the peptides. And the last one is the sample preparation. The sample preparation is not straightforward as like small molecules. We may see a mix of two techniques to get a cleaner sample when we are working for proteins and peptides as well. So the major challenge, as I said before, we face in the peptide quant is the preparation of cleaner sample for LC-MS/MS analysis. And we have literally seen that the scientists spend months of time to optimize the sample extraction method for peptides. And there are three types of sample preparation techniques are available in general. The first one is your protein precipitation. Simply crush out your protein components from the biological fluid, leaving behind the peptide targets. And this may happen either by adding the organic solvents like your methanol, acetonitrile, ethanol, IPA, etc., are also even precipitate by adding the organic acids like TFA, TCA, etc. But the important thing to consider here is the stability of your peptides in the acidic condition while you are precipitating with the acids like TFA or TCA. And the second one is the solid phase extraction. And the principle is selectively you isolate your target peptide of interest while removing the matrix components. And this is widely accepted in the industry as well. And these SPE extraction technique in today's market, you see the lot many cartridges are available with variety of phases and sizes. And this should be carefully chosen for your sample or your analyte of interest. For instance, you have reverse phase or ion exchange SPE. There are polymer based as well as the silica based SPEs as well, or you can choose your well plates or the microelution plates. But before choosing that, the important thing to consider is the recovery, which is influenced by the type of pressure you apply during this XP extraction, like your positive pressure or negative pressure. Many times we see that for the small molecules, either the positive or negative doesn't make a significant difference, but when it comes to peptides, definitely it makes a lot of difference when it comes to recovery. And the third one is the affinity capture. And there are two subtypes. Either it could be an affinity depletion or an affinity enrichment. And here the requirement is you need a very specific antibody. And we don't generally see this kind of affinity capture based cleanup when it comes to peptide quantification in the biological matrix. The second most challenge I would like to discuss here is the low level quantification. The quantification at low level, like your sub-nanogram or sub-picogram, would be the primary requisite for the peptide bioanalysis. And a high-end triple quad system, which offers sensitivity and robustness in the biological matrix, is the mandate for the peptide analysis. We have, or you might also been seeing that many times the sensitivity would be good when you inject it as a neat solution, but the sensitivity requirement at the matrix is really questionable when it comes to these proteins or peptides in the biological matrix. Really, you need a high-end triple quadrupole system for this kind of analysis. So SciEx 6500 plus is the workhorse for this peptide analysis. And what made it to be the optimal MS for the peptide quants? The SciEx triple quadrupole 6500 plus system features the multi-component ion drive technology, including the ion drive source, your ion drive QJet, and the ion drive high energy detector plus. That really pushes the boundaries of the LC-MS quantification farther than ever before. Just a brief note on the technological advancements in 65 plus. The new design of the legendary Turbo V source increases the ion production by enhancing the gas flow dynamics as well as the optimized heater configuration to improve your reproducibility and robustness. So what you see in the 65 plus heater is much larger than the traditional 4000s or 4500 base systems. And also with its dual stage design, the ion drive DJet guide really improves the sensitivity by focusing the ions more efficiently towards the Q0. And also the new detector system on the 65 plus has two main improvements. The first one is the high energy voltage. We increase the voltage from 10 kilovolt to 15 kilovolt to provide an improvement primarily at the high mass. So as we see the peptides, the masses are going to be on the higher side. The HED detector plays an important role to offer you the best sensitivity. And also the system comes with the floating detection system that really improve the negative mode sensitivity as well. The negative ions are now directly focused to the detector, bypassing the HED conversion. The deflector conversion of ions to electrons is completely neglected here, and the ions can directly reach your detector, thereby increasing the sensitivity, particularly at low mass. We would also like to highlight some of the methods that are developed in this 65+ system. So you might have the question, probably you have brought in a lot of ion drive technology. So how it is getting translated into the results. So here is an example we would like to highlight here. The number one is the exenatide. The exenatide estimation in the human plasma. And exenatide, as we all know, is a human GLP-1 analog with a molecular weight of 4186 Dalton, and is the first member of the class of GLP-1 receptor agonist as well. And literature says that the exenatide has a longer half-life of about 2.4 hours compared to the glycogen peptide as such. And these are available as the subcutaneous injections or even intramuscular injections. So the literature suggests that following the subcutaneous administration of a 10 mcg dose, the Cmax observed was around 211 picogram per ml. And there are also the doses available at 5 mcg as well. So that clearly indicates if the Cmax is around 211 picogram per ml, and what should be the LOQ, that really needs a very, very high-end instrument for this quantification in biological matrix. So SciEx, we have developed and qualified a method on QTRAP 65+ system to achieve the LLOQ of 19.5 picogram per ml. And what you see is on the MS parameters and the LC conditions as well. I'm not going to go through that. These are self-explanatory. And these are the representative chromatograms, what we have achieved for the exenatide in 65+. The sample extraction, we have applied a very, very simple sample extraction technology. We just taken the aliquot, pre-treated with liquor ammonia, passed on to the anion-based cartridge, dried, reconstituted, and injected for LCMS analysis. And what you see here is the blank sample. There is no significant interference at the retention time of your analyte. And also the LLOQ of 19.5 picogram is very sharp with the S/N of 28. And considering this intensity, we still have the scope to further go down when there is a need. And this is the chromatogram for the ULLOQ at 1250 picogram as well. So this is the linear response, which we observed across the concentration range of 20 picogram to 1250 picogram. And we also see an R value of 0.99, absolutely meeting the requirements of the regulatory guidelines. So the next method would like to highlight, which we developed in the 65+, is the liraglutide and semaglutide, again in biological matrix like human plasma. And just a brief background on the liraglutide and sema, and both are again a GLP-1 agonist, but with the optimal fatty acid and linker combinations. For instance, you see a C16 side chain for liraglutide and C18 for semaglutide. And these has been identified to maximize the albumin binding while still maintaining the GLP-1 receptor potency. And semaglutide is a once in a week injection or in a once in a week injection, and liraglutide injection has to be given every day because semaglutide has a longer duration of action in the body. And what the literature suggests is following the subcutaneous administration of just a 0.6 mg of liraglutide, the Cmax observed was around 35 nanogram. And semaglutide has a long-acting peptide. Just an administration of 1.34 mg, it slowly get absorbed into the system, and the Cmax was observed between 24 to 122 hours. That significantly indicates that how your method should be robust, and also to properly find out your elimination rate constant, how low level LOQs are really required in your methods. So these are the MRM conditions and the LC conditions we have used for the SEMA and LIRA in human plasma. And as you see here, these are the Q1 masses. These are the Q1 masses for LIRA and LIRA as well as for the SEMA. And what unique thing you observe here is we intentionally chosen a Q3 mass higher than the Q1 mass, just to avoid all your interference from these singly charged peptides. And all these parameters are automatically optimized by the software. And one important thing to highlight over here is the source and gas parameters in the SciEx systems. We use a cut-in gas interface, unlike a capillary interface, and also the temperature is the only one point of applying temperature in the SciEx machines. We don't apply temperature at multiple points. There's only one point. That temperature is supplied by your GS2. That helps really while you are optimizing the samples in a much faster way. And also would like to highlight the results, what we observed for the SEMA and as well as LIRA. And the sample extraction methods, we just kept it very simple. We had just taken a 200 microliter plasma, we precipitated it, taken the supernatant, dried and just injected into the system. No concentration step is required because the machine is very much sensitive and also the reproducible. So these are the representative chromatograms, what you see on the left side. An LLOQ of 200 picogram per ml with S/N of around 100. And also the carryover, as I said before, these peptides have the tendency of absorption, which may give to column carryover or the autosampler carryover. But the SciEx team has come up with a method with no or negligible carryover, or even after injecting 1 lakh picogram per ml, you don't see any carryover in the chromatographic run. And also the detector, as I said before, the HED detector has given a very good linear dynamic range from 200 picogram to 1 lakh picogram per ml. And also on the top, you see the statistics showing a person CV at 200 picogram per ml less than 7. That is really interesting when it comes to the peptide quantitation in biological matrix. In the same way, these are the representative chromatograms for LOQ at 100 picogram per ml for liraglutide. And what you see is the S/N of around 150. So still have the scope to go beyond this LLOQ as well when there is a need. And one of the challenge while analyzing the peptide is the possible contamination of the ion optics. If you are continuously running the peptide quantification, you may come across this challenge, the contamination of the ion optics due to the high load of matrix interference. And we observe sometimes in certain cases, you may force to use the BSA or even some surfactants for your sample preparation. And literally that may tend to contaminate your system frequently. So how to mitigate this issue? And SciEx has really come up with the scheduled ionization, that is the ion spray voltage, scheduled ionization ISB. So what exactly is the source voltage will be turned off other than your analyte retention time. So if you refer the figure here, this is your start of the chromatographic run where you are expected to elute this phospholipids, and after some time you may also expect it to elute more matrix interference as your gradient steepens, your organic content steepens. So probably if this is the retention time of your analyte, the ion spray voltage will be applied only at this point, and rest all the time the ion spray voltage will not be applied. So as a result, your mass will be kept clean, and it's very much stable to go for a long period of time. And this is a very perfect example which we have applied this ISP in the IGF-1 and IGF-2 quantification. So in this sample preparation, we are expected to use chaps, that is again a surfactant in the sample cleanup to minimize the protein binding of this IGF-1 and IGF-2. And even though we can remove majority of these chaps in the sample cleanup, but still some amount of traces were always there in the sample. And what we have done is we have chromatographically separated this from these analyte of interest, and at this point of time the ion spray voltages were switched off. As a result, this will not go and contaminate your system. So based on this software feature, we have developed the method at 2 nanogram, perfectly meeting the regulatory guidelines for these kind of complex molecules as well. And also sometimes you come across certain challenges while doing the peptide quantification. These peptides doesn't fragment well in, especially the cyclic peptides, right? And this low fragmentation efficiency typically translates to the poor sensitivity. And the classical example is the somatostatin, and look at the MS2 spectrum. There is no predominant peak to get a better quantification. And in general, we use the MRM as a technique for MRM scan is the technique for the quantification. But in this case, since there is no predominant MS2 spectrum, MS2 ions, we cannot use the MRM. It may lead to poor sensitivity. And what we have usually see is, instead of MRM in this case, the literature suggests, or many labs may try to do the selected ion monitoring or a single ion monitoring. However, we know that this SIM may lacks in selectivity at many times, right? And what we have come up from SciEx is we used an MIAM workflow, that is a multiple ion monitoring coupled to a SelectIon Plus device. That's a differential mobility separation to achieve the low level selective quantification of this somatostatin. So what it indicates is, in the parent and on the daughter, the Q1 and Q3, we monitor the same ion, and even though it tends to give a selectivity issue, but the SelectIon, the incorporation of the SelectIon would definitely gives a next level of selectivity in these cases. And the very important to notice noted down here is this also happens at the MRM scale. So SelectIon, the differential mobility separation technology, you can very well use at MRM scale, very much suitable for the quantitative experiment. So a picture at the bottom really depicts the SelectIon components, which comes with the 65 plus system. But again, it's an optional. When it is required, you can use this instrument for certain complex peptides where there is a poor MS fragmentation. So this is the one I would like to show on the results. If you see on the extreme left side, this is the MIM workflow without SelectIon. You are monitoring the same ion as your parent and your daughter, and there is a high baseline what you see. But at the same time, when you use this SelectIon in this MIM workflow, the baseline has significantly reduced, and as a result, even a 50 picogram per ml, you can see a very good peak, giving a percent CV of less than 6% at 50 picogram. That's really interesting when it comes to the rat plasma matrix. So a new era of precision bioanalysis, as I discussed before, in spite of the 65 plus being the highest sensitive machine available in the market, and still SciEx is committed in advancing the scientific research through its precision technology. And the SciEx 7500 was recently introduced. I presented sometime before as well. And from there on, it continuously surprised many of our customers. Right, the SciEx triple quadrupole 7500 system is designed with a new source for better ionization, all new ion optics, and as well as a HED detector that enables a new levels of quantification of proteins or peptides in your labs. And the new ionization source in the 7500 is called the OptiFlow Pro ion source. So what is this OptiFlow Pro ion source? It carries the same legacy of the Turbo V source, which we introduced way back in 2001, but with an additional component called E-lens technology. And to be noted, the OptiFlow Pro ion source is an all-in-one versatile ion source. As you see over here with the interchangeable towers and probes, so easily you can switch between the analytical flow and the microflow. The source is same, but you can easily switch between analytical or the microflow by changing certain towers and probes. And this is the source that can support up to 3 ml per minute. And that's the only source you see in the market. And as I said before, this E-lens technology, what we have incorporated in this source is both for the analytical as well as for the microflow, but only for the ESI mode of ionization. So what is this E-lens technology to us? Why we have come up with this E-lens technology? And as you see here, consider this is the tub of his source housing on the left side. And as the ESA droplets are moving towards the orifice, at certain point of time, you may see that an amount of ions may always go into the exhaust. But the ELANS technology creates a stronger field inside the source to drive the ions more towards the orifice. So this really helped in performing or in doing the desolvation within the source, and you see a twofold improvement in sensitivity when you are incorporated with this ELANS technology. So you might ask that, okay, you have brought in a lot of technological advancements in the 7500 system compared to the 65, which is already a most sensitive machine available in the market, very much suitable for the peptides. So how is that translating into the, how it is getting translated into the your samples? So what else change has been made? And since there are more number of ions forming in the ionization source, we have enlarged the orifice as well. But to incorporate this enlargement in the orifice, the D-jets are also incorporated. The Q-jets alone are replaced by D-jets followed by Q-jets. What you see here is the D-jet, where a dodecapole structure followed by a Q-jet is incorporated to give more efficient transmission of ions from the source into the system. And because of these technological advancements, like your difference in the source, your orifice is larger, and as well as the ion optics is different, like Q-jets are replaced by the D-jets, there is definitely a sensitivity gain in the biological matrix. So to evaluate that, we have set up an assay on 65 as well as 75 for a few molecules like propanol and penpyroximate. And what you see here is the absolute area gain of 9x compared to that of the 65 plus. And not only for these two molecules, we have done more than 1244 MRMs, both in positive polarity and negative polarity across multiple studies like your pesticides, drugs, or even for peptides. And the average peak area gain what we have seen in this mission is 7x compared to 65 plus. Right. So we will also highlight to some of the methods what we have developed in the 7500 system. The first one is the natriuretic peptide. These are the peptides, these are the cyclic peptides known for poor fragmentation, and many times the literature suggests people go for the single ion monitoring, right? But it lacks the selectivity. We have taken that as a challenge. We have taken these molecules like ANP and BNP. We precipitated the rat plasma. Then we have passed on to the mixed mode SP cartridge. The elements were just diluted, and we analyzed both in the 65 plus and 75. And what results I'm going to show is really astonishing. This is a wonderful improvement in the sensitivity. What you see here is the 5x improvement in sensitivity both for ANP and as well as for the BNP peptides. And these are cyclic peptides known for the very, very poor fragmentation. So this is the linearity curve, what you see from picogram, 50 picogram to 100 nanogram, and the statistics also plotted on the left side. And you see these are really, really good when it comes to the cyclic peptides in these missions. And this is the last slide I would like to show for the 7500 performance. And this is once again the insulin Lispro quantification using 75. And those who worked in the insulin might be aware of how difficult, how challenging it is to get the LLOQ in the matrix. There are multiple reasons. Again, this is a cyclic peptide with disulfide bonds. And what we have done is the samples where the protein precipitated, the supernatants were collected and processed by the mixed mode SP. And the elements were just diluted with water and injected into the LC-MS analysis. And what we see is the 4x difference in sensitivity between 65 and 75. And also I'd like to convey an important message here is probably might be working on a 55 or 65, and you probably the 65 itself might be sufficient for your level of sensitivity. But what the 7500 can offer you is the simplification process in your sample extraction strategy. Really, no need for you to take a much higher aliquot or to concentrate the samples to get the required sensitivity. As such, it is offering a 7x improvement in sensitivity. You can very well precipitate and then dilute, or dilute and shoot method will very well work for the 7500, even though you're not looking for a very high level sensitivity in your lab. Right, so this is the summary of the 7500 system, really the DJIT iron guide, what we have incorporated in the system, along with the integrated E-lens technology. That is the first of kind technology we have incorporated in this 7500 system, combined to deliver the ultimate sensitivity. And what we see is the average peak area gain of up to 7x compared to the high-end machine like 65 plus. And the signal to noise in general, we see a gain of 2.5 to 3x compared to 65 plus. And the source is also in this system is also an important parameter to talk about. It has the reliability to easily, it has the reliability as well as the efficiency of the legendary turbo V source of the SciX machines, right? And the new modularity, what we have brought in it helps you to easily switch between the high flow and the low flow or the microflow in a matter of minutes, right? And the detector is also up to 6 orders of magnitude. That really helps you to cover a wide range of analyte concentration. And as I said before, the source is different, the ion optics is different, and that really brings you an uncompromised robustness for your peptide analysis as well. So this is my summary and conclusion. We know that the peptides really have attracted a lot of attention in the last decades, right? And the number of approved peptide on the biotherapeutics, the peptide biotherapeutics has been on the rise in the last decades, and more than 150 peptides are under development. And if you're working on a small molecule, the major challenge what you see in this quantification is to achieve a low level of LOQs compared to any other small molecule analysis, considering its high potency and also the requirement of, and sometimes there is a requirement of an additional dimension in the selectivity as well. As I said before, the peptides which are cyclic, which are having a poor fragmentation efficiency. And in the 65 plus is the really in the mass spec choice for the peptide quantification because of its ion drive technology, which really supports the scientists to achieve the LLOQ of most of the peptide drugs. And also SciX offers the SelectIon, that is the differential mobility separation technology that helps you to reduce the matrix interference as well as the background noise originating from your isobaric compounds present in the complex plasma matrices. And also the 7500, what we recently launched with its innovative DJIT ion guide and the integrated E-lens technology, combined to deliver the ultimate sensitivity for your peptide analysis. So we talked about the 65 plus, we talked about the SelectIon and as well as the 7500 system. And I have shown some of the data what we have generated in our lab to showcase the instrument performance with real life samples. And with this, I'm concluding my presentation. Thank you so much for listening and open for questions.
Great. Thank you, Jan, for bringing this new technology to all of us. Let's see. What I would like to do is we will call upon others speakers as well, so we can have the questions. There are a couple of questions for you, but we'll take it up as a part of panel discussion. I see Arjun is already there. Thanks, Arjun. Can I request Aparna, if she is around, to join the panel?
I think Aparna is
Can
there.
you hear me? Yes,
Yes,
I'm here.
Yeah,
Hello, can you hear me?
Yes,
Yeah, I can
Aparna.
hear you.
Yes, there I am. Okay, thank you.
So good. We have all the three speakers except Darshana, and thanks for staying back. We have a couple of questions popping in from the audience. So let me start with the question for Jan. Jan, Shrikant needs your inputs, and he is mentioning about more the sensitivity, more the chances of contamination. So earlier days, when API 4000 used, people used to open the mass rail, and that was a big task, right? But higher the sensitive instruments, the frequency of mass rail access because of contamination is going to be a challenge. Can you throw some light on maintaining the equipment when you are talking about higher sensitivities?
Yeah, so definitely that's a fact. If you're walking the lab, definitely you come across this point. Higher the sensitivity, higher the chance of contamination of your mass spec. Right, but there is a need in the market, and the vendors has to come up or they improve their mass detection level. So what we are doing, and if you typically take the 4000, the ion optics starts with the skimmer, but later the skimmers are replaced by the QJets, and the QJets are replaced by the DJets as well. So, so from the instrumentation side, right, the instruments are coming up with a much robust, rugged hardware. We are replacing the old technology with the newer technologies on the ion optics to reduce the contamination. But in spite of that, yes, probably with respect to your lab conditions, your solvents, these are tend to contaminate the system compared to the low emission. So special precautions are really required with respect to your sample cleanup, as well as the solvents what you use to have a rugged system in your lab. Right, so these things are taken while we designing the instrument, but of course there is a need for the laboratory scientists also to take care of the instruments, especially when you're working on the high-end systems.
So, Jayant, this is Amarnath. In line with the same question, what the other folks has asked you the question about the contaminations. What's your experience? What kind of the precaution to be taken in terms of avoiding a contamination with the 6,500 plus system? I mean, it's a practical experience. We have also seen the frequent contamination of the mass spectrometry system, in spite of taking a lot of care with the systems, even in the isolated room of being made for this instrument. So what's your experience? I mean, can you give some thoughts on this? What kind of the solvent to be used? What are the precautions to be taken in terms of avoiding a frequent contamination of the system and the breakdown of the instruments?
Thanks. So out of my experience, I would say that the first and prior most thing you need to look is the solvents, right? The quality of the solvents. Probably if you really want to evaluate the quality of solvents, you take some amount of solvents and do a Q&A mass, and you can see really how many of the high intense masses are there in your solvent, and that's really going to contaminate your system. But that is on one aspect, right? And this that is on one aspect. You always are recommended to use an LCMS grade solvents, and that has to be without any much interference when it comes to small masses. You should not give contamination to the system. And the second one is your sample cleanup, right? If you're just precipitating the proteins, but how about the other fats and other components which are there in the plasma, whether this is interacting with your mass system, that you really have to have a look. And the third one is the ion spray voltage, right? We have come up with the ion spray voltage, the scheduled ionization voltage to bring down the contamination issue when you are using the high-end instruments. So definitely you can look for optimizing the software features for your methods as well to bring down the contamination.
What do you feel about use of the gradient grade solvent instead of using the LCMS grade solvent? Because that is again the high purity the solvent, though it may be a 2-fold, 3-fold expensive, but to my understanding, if somebody is using the gradient grade solvent, where they can also minimize a lot of background, lot of the traces material as well. So I mean, we just started using the gradient grade solvent, and we have seen that up to certain extent, this can avoid the contamination part. I mean, even it's a lower end instrument. Even API 2000 people used to use the LCMS grade solvent. But I just wanted to your thought on the use of the gradient grade solvent, which is very, very expensive, no doubt.
To be honest with you, sir, I'm really not aware of the gradient grade solvent, but what in general we use in our lab is the MS grade solvents, right? And it's not that every time or for every molecule your system is going to get contaminated. We have seen in many places the 6500 kind of instrument, the high-end instruments are running day and night without contamination. But the challenge would definitely come when you are looking for proteins or the peptides kind of quantification in the missions. But definitely, I would say if you're improving the quality of your solvents, it may improve the life or it may decrease the downtime. But at this point of time, I'm not really aware of the gradient grade solvent and what is the quality checks and what kind of, how it is better than the MS grade solvents. I'm not aware of.
Okay. We have just one, the quick question. It's not question, just wanted to know in terms of the exenatide, what is the plasma volume used?
So exenatide we have done in the human plasma, right? So in general, we use, if it is a human plasma, if it is a 200 or 300 microliter aliquot volume. If it is a rat plasma, we use a 10 to 25 microliter. But at this moment, I do not have the exact volume with me. But if it will be somewhere around 200 or 300 microliter. But if you want a definite answer, I can come back to you.
Sure, there is. Yeah.
Go ahead, Sunny. Go ahead. No. Just related to the exenatide itself. Yeah. Have you done any assay selectivity in terms of, there are two metabolites reported for exenatide, though it's not necessarily required to be estimated, but it's mandatory to the show your assay selectivity. Have you done it, those metabolites?
Not really, sir, because from an instrument point of view, vendor point of view, we always try to establish the maximum sensitivity of the machine, and it's sometimes it's based on the requirement of the customer as well, right? And if there is a requirement where you wanted to separate using the chromatographic conditions, definitely we do that. But in case of exenatide, our objective was to establish the sensitivity, not really on the separation between metabolites.
Okay, no problem. I'll catch you, Jan, offline, because I need some information on exenatide as we are working on this product, and I know that the challenge of this product is I don't know if some of the lab has the method readily available, but it's really a challenging complex product. I'll catch you in offline.
Definitely.
Go ahead, Srini.
Yeah, I was just seeing one more question popping up. This is for Aparna. This is again Shrikant. He's mentioning that in pandemic, the internal and external audit preparation has been a whole new process. Can you share thoughts on advantages and disadvantages of these audits in this pandemic time? Both. You might have physical inspections or audits or online inspections.
Disadvantages and advantages. I mean, I'm not sure I understood the question very well about.
I mean, which is the preferred way?
Yeah, which is a preferred way? Advantages. Okay, of course, at any given point of time, a physical audit is the most preferred because there's really nothing that can compensate for the actual physical interaction that we have. I mean, the face-to-face interaction that we have, the body language that we keep with auditors. There's no compensating it. But having said that, we have been quite successful in our online or our online assessments, especially with external auditors. So the key points about online assessment, I believe, is the pre-audit preparation, right? So it's very important to have a clear agenda, and it's also very important to have mock trials with camera as well as voices. And we found something that was really helpful for us is to make a virtual tour with the auditors as a part of the pre-audit preparation. So the auditors really know about our laboratory, know about the people, and know about the layout, etc., etc. So it helps them kind of, when they're reviewing the SOPs, it gives them, it makes our processes a little more visual, I believe, to them. On the days of the audit, what really helped us is good quality cameras, because people want to speak, people want to see what's happening on the bench with and who they are interviewing and our responses, etc. So good quality voice and camera is extremely important. We increased the number of audit rooms, so we had not more than 2 or 3 staff in each room, and ventilated rooms, and all staff with non-MS teams. And of course, all our documents were shared through e-room. One thing that really helped and worked for us is that the auditors, especially the auditors from US and EU, as well as our Indian auditors, client auditors that we had, would do this staggered auditing. They would take some time off and review, say, for the first few hours where we didn't have to be present. We just have to give them documents and be continuing with our own work. And then they would call us for interviews. And again, there would be a review time and then an interview time. So this way, there was a lot of time both for them as well as us. And frequent breakout rooms were created where every now and then auditors would come in and say, hey, we have some questions. And we have had ad hoc meetings and ad hoc slots. So it pretty much worked very well. Of course, as I said earlier, it can never compensate for face-to-face interaction, but it did do a lot in terms of bringing advantage in getting our laboratory qualified and validated in spite of a lockdown or a pandemic situation. Again, with all audits, at the end of the day, being transparent, being honest, and being clear in communication. Whether we are taking an offline or an online audit, these are 3 very important key attributes of how we want to face an audit. I hope I was able to explain a little bit
Yeah, I think
on that point.
Yes, you clearly mentioned in terms of auditees stand. And let me just add auditors' viewpoint as well. Most of the times, the auditors are also not comfortable on remote audits because it's the only data that speaks. Though you give virtual tools, those are momentary. And they also miss a 360-degree look at what's happening on ground. So, but yes, given circumstances, that is the best option available. And that's how auditors are also trying to do their best to adjudge that the data is reliable and can really go ahead with that. So
in fact, I'm sorry, I just wanted to add, Dr. Savli.
Right.
In fact, when we were being assessed by NABL, during the lockdown time for RT-PCR laboratory, they ensured that there was 1 person that was available on site, and then there were 4 that were online. So that way, this person that is available on site was really the liaison, to the rest, and was a connectivity between us and them. So nothing is really lost in translation, over the internet. So that also helps sometimes, because we can afford to have 1 person on, out here, and if they're willing to travel. So.
Yeah, I think that's what even USFD used to do. Though a couple of inspectors are on ground, they have a backup team which always has been looking at the day-to-day data exchanges. So good, thanks for that point. We can call them as hybrid inspections. So coming to Arjun, Arjun, you were talking about the large molecule PK analysis and assay developments. Any thoughts on the ADCs, the antibody drug conjugates? How the strategies would be a little different than a typical large molecule?
Yeah, I don't have that much experience with ADCs, but I think the procedure will be the same. I mean, you also have critical reagents. Only if you have an ADC, then it's not like an antibody that you have like 2 the same anti-IDO types. Then you have different approaches. But for the rest, it would be the same. Yeah.
Yeah, especially
maybe some ADCs can be a little bit less stable. Normally, most of these projects are all irregular antibodies. And that is, actually all these PK assays are a bit the same. But I also have like yeah, other kinds of proteins, and then you might end up with you know, other results. I cannot speak, of course, about, you know, the actual product. That's like not allowed. But I know from, for example, there was some product in which we, you know, set it up, the assay, had a nice curve, at least we thought we had. And after a couple of development experiments, it seemed that in the top of the curve, there was some wiggling going on. And first of all, the technician thought, well, you know, these are the first runs, and I just didn't pipette that clear. But it turned out to be actually a feature of that drug. So you have to look very carefully to your data. So it ended up that it meant that the ideotype pair was not suitable. Because, for example, if a molecule becomes very small, then you can have that at the upper part of the curve that you have interference, that you have like that's you know, the molecules are small, and you can have some weird kind of responses. So what I would advise, if you have such a drug, that look very carefully to your data, do a few runs, and be careful what you see. It might be that your result, what you see, that is just not a matter of not good pipetting, but can also be that something happened with that molecule. For sure, you can see that. And also you have like, with ADCs, you might have stability issues, which are different from your regular antibodies. So you have to be careful. What I always say is look at the data. Look carefully at the data. And don't follow a protocol, a guideline like, okay, we are going to do this step and that step. You have to look at the data. You are a scientist, don't forget that. So be careful. Yeah.
Yeah, Aparna, you wanted to add something?
Yes, you know, we don't have too much experience with ADCs, but, you know, talking about ADC type of molecules, whether it's a conjunction or talking about bispecific antibodies, and, you know, I would like to echo the same thing Arjun said. The most important aspect is looking at those 2-3 different parts of this bound and of this one molecule, and look at critical reagents that are going to be specific. For example, you're looking at bispecific antibodies. You're looking at determining immunogenicity of a bispecific antibody. You know, you really need to know where is the cusp region. Is that going to be immunogenic? Is arm 1 or arm 2 going to be immunogenic? And you have to really think and design your critical reagent well before you really start working on the assay. And the mistake that bioanalytical scientists usually make is that we concentrate too much at the lower end of the curve. We're always looking for sensitivity. We're always looking for signal to noise, where sometimes the way that the curve moves, in the upper end is something that we don't see. And that also, we should draw our attention to really how the curve moves and how the antibody actually responds to the top part of the curve. That's something that we which will help us in bispecifics as well as ADCs, and as therapeutics get more and more complex, I believe.
Yeah, I agree with that. I agree with this.
Yeah, Srinivas.
Just quick comment.
Am I audible, Srinivas? Dr. Ravi here.
Yeah, Ravi, go ahead.
Yeah. So so as Aparna and Arjun touched upon the critical reagent, so I have one question on that. So generally, Arjun has nicely covered the critical reagent management in terms of kind of a life cycle management. So that's really a crucial part of any kind of a bioanalytical as well as bioassays. So considering these aspects, at any point of time, due to unfortunate situation, if we have to altogether change a critical reagent, antibody and ligands or any kind of critical reagent. So what would be the expectation of the bridging study? So when we look into a guidance, so it's bit broader. So from the experience perspective, probably Arjun or Aparna can add on. So what would be the regulatory expectation on full length? And if the sensitivity would change altogether, then what kind of justification may expect from the regulatory?
Okay, so yeah, bridging experiments, that's actually your question. How should you bridge a new critical reagent? Well, there are also white papers on it, how to do it. But if, for example, if you have to change your your capture antibody or your detection antibody, it changes or whatever, it's a new lot, and you don't see the same response. It's not a problem that you just tweak your capture concentration, your detection concentration. You don't have to keep that fixed. It's not an issue that you that you change your so as long as the critical reagent is the same, but you have a new batch, lot batch, a new production batch, it's not an issue, and you have to do a new labeling. It's not a problem to just tweak your concentration. It's not an issue as long as the curve with your drug is the same as as in the previous assays. So that is not a problem. Does it answer your question or not?
Yeah, partially. I mean to say sometimes they ask kind of impact on the previous batches analysis, I mean to say. So that was one of the issue what we have faced. So in such situation, kind of like in case of cell-based assays, we need to continue this for 6 months or a year, something like that. So I just wanted to know from your opinion.
Yeah, okay. You will bridge in your previous batches in your assay, and that should be the same as before you change the reagent. That is something you should do. And
okay.
Yeah. That is not something you have to make a problem of. For example, if you have a new reagent and you have to do a new labeling, it's not unusual that your labeling is not 100% the same as the first labeling. So that changes a bit. It's not a problem, as long as you still meet your accuracy and precision criteria, and you should also meet that the LOQ and the ULOQ. That should be the same. If your curve completely changes, that's something else. But no,
yeah. So Ravi, the luckiest. You're really lucky if if nothing changes between your critical reagents, right? If you're going to be changing either a lot or the label or any other concentration related parameter. But if something does change, of course, to know that it's changed or not, we in the laboratory we do what is called as a reagent qualification, where we have the old lot and we have the new lot, and we compare it based on validated acceptance criteria. And if we don't have the historical lot, then we use historical data to see if the label, especially large molecule critical reagents, are aligned or not. But as per the bioanalytical method validation guideline guidance document from the US FDA 2018, the FDA is asking for some kind of partial validation or assay validation, excuse me, when there are critical lot-to-lot changes or any other type of change which will impact the data. So we should always remember, when we are changing a critical reagent, we should be aware and sure that we should put in some kind of a partial validation right there to make sure that the data is not impacted. There have been certain cases where incurred sample reanalysis has been performed with the new lot of critical reagents, just to check that the data does not move between an assay with the old or the new reagent. That is also allowed as long as we have justification. So there are many ways and means of proving that the assay or the data is not compromised. And I believe while we follow guidance, we should also use our own intelligence and common sense to see that we want to cover all gaps for adequate traceability and continuity of reagents, Ravi, especially when it comes to cell-based assays.
Yeah.
Amarnath, would you like to echo? There is one question for Arjun, question and answer box.
Sorry. Is there a question for me?
Yeah.
Yes, Sergeant. I think.
I see. I see them.
Okay.
So I have a question regarding under what situation ELISA and MSD are.
yes.
Yeah. Yeah, I would say, why just use MSD all the time? Because that's like the preferred. ELISA, to my opinion, is a bit old-fashioned. Maybe some companies still do ELISA because they want to have those assays the same as in the past, and they do that assays for decades, then they still use ELISA. For example, I've seen years ago that people used a very old-fashioned detection system to determine some antibody against, I believe, against flu or so, because they already did that for decades. But you know, with all these new products, I would say use the MSD platform, because that is like the platform which actually is used all the time for everybody. So every regulator would understand these data. It depends, of course, what you kind of assays you want to have. If you want to have a very sensitive assay, yeah, you have to explore new platforms like Simoa, for example. That is something we are going to do now. That's quite a new platform. So you might consider. You just need to find the platform which is most suitable for your whole assay. And for example, if you have also links to your study. So for example, if you have study with the children, you know, very small children, the volume of your sample will be very low. Does that agree with your ELISA? I don't think so. Maybe you should go to GyroLab, because that is requiring only microliters. So it depends what your aim is. But yeah, ELISA, I think I would only do that in very special occasions where you cannot do anything else. It's yeah, that's my opinion.
Thanks, Arjun. So with the keeping interest of time in mind, can I ask Deepak to display the certificate for Jen? We would like to appreciate his contribution.
So you are showing so many certificates, Deepak.
Oh, is it?
Just click one which is relevant to Jan.
No, I think it has taken a different window. Just give me a moment. Yeah, I think now you are able to see.
So Jen, formally thanks a lot for your contribution to this session, and kindly accept the certificate of appreciation from APA Organizing Committee. Thanks all of you, Jen, Aparna,
Thanks,
Arjun,
Srinivas. Yeah.
for your contribution. And
So,
I'm sure there are many questions, but we will keep interacting and connecting offline.
of course, I
Over
would
to you.
would request all the participants, if you have any further questions, feel free to post it on bostondb21@gmail.com, and we will reach out to each of the speakers and get your doubts clarified, or further information on if you may need on any of the topics which we have discussed today. So before I request the moderators, Amarnath and Srinivas, for their concluding remark, let me quickly launch the poll. So for all the participants and the panelists, you will see just 3-4 questions on your screen, and that's basically the poll. So do submit your response. I think it's already been launched, so it should be on your screen. And while we take up the concluding remarks, you can please submit your response to this webinar poll. Okay. So with that, first of all, would like to thank all our distinguished speakers, starting from Aparna, Arjun, and GR, and also Darshana. Thanks a lot for your time and a very interactive session we had, and very informative talks which you delivered. And hopefully next year we'll be having an in-person meeting, and looking forward to have you, all of you, in person delivering the lectures at APA. And I think it's been a long journey. I think this is our 13th webinar. So we have been running since last 5 months. So I know for moderators, even for the audience and for the speakers, been very exhaustive session. But yeah, very interesting, because people have really committed to this, and they've been attending all the webinars, all the talks. So we really appreciate your active participation, all 3 of you, and looking forward to have you back again next year, hopefully in person. So with that, let me request Amarnath first, followed by Srinivas for their concluding remarks. Over to you, Amarnath. Thank you.
Thanks, Deepak. And in a single word, today was really an excellent talk. All the people have covered the different corners. Like the Aparna, she covered, how she could able to manage, both the productivity and the balance between, the implied to. I mean, it's not too easy to manage such a, the pandemic situations, because nobody has such a kind of the bad experience in the life. So it was very difficult how they can manage, the lab productivity, managing people, and the most importantly, retaining the people, the employee, the retention was very, very important during the this period, because at the same time, the management also need to have the productivity, the productive job, and, the run the business there. So I think it was really a critical situation across the globe, across the lab, and the difficult situation for only the pharmaceutical industry, because everybody has to work there. They were the only the people who has not shut down their business. They have to go, took the permission from the government, and wherever the possible, people have to be there into the lab. Without the lab function, the people can't review the data sitting at the home. So it was a really challenging work, and I think majority of lab, they did a the great work. And that is what, the Parna took a very good talk today, and how she managed, the across the lab functions, arranging the people who will sit in the home, who will be there into the lab to make sure that everything works smoothly. At the same time, I think I told Arjun that he has again the refresh the mind and the guidelines, the development approach, the validation aspect as well. And Jan, I mean, it's always good to have, the support from the psych teams. I mean, they really do the good job, and took a lot of the challenging product. I mean, many of the times it's not practically possible for the lab to spend a lot of time, because they have the limited resources. They have the lot of the workload on. And in such situation, the psychs always support for the application, the work. Like in this case, they have done a very, very, that they took a very complex product, all the liraglutide, semaglutide, and the exenatide. All the three products are, because I'm working on these products, I know the complexity of these products. And this kind of support is really appreciated from the vendor side. They used to support for such kind of the complex molecule, and that helps a lot of minimizing the method development approach. I mean, one can directly take their method just to re-optimize in their lab, and then straightforward they can adopt the procedure and do the validation part. So I think that today the topic which Jan has taken, all these three products, is really appreciated. And as I said, Jan, I would offline catch you and understand more details on this product. But overall, the session was really the excellent talk, and the good discussion with all the folks. And I'm happy to see even the Parna after a long time. So it was a good interaction. So overall, thank you very much for all the speakers. And unfortunately, Darshana was not able to attend it, but probably we'll see her in the next year. Thank you very much, all of you.
Thank you,
Thank you.
Aman.
Thanks.
And finally, before I request Srinivas for his concluding remarks, I would like to mention to all the participants that the poll is still on. So you can just click on the polls on your screen. There are a few questions. Please submit your responses. Immediately after Srinivas's concluding remark, we will do the Kindle draw. So over to you, Srinivas, for your final 1 or 2 minutes concluding remarks. Thank you.
The 13th week, right, Deepak? I think we are like really growing with science as well. So we had biotherapeutics as theme this time, and excellent talks. All the 4 talks, even Darshana touched upon the complex bispecific antibodies and strategies around that, followed by Aparna's in-life experience with managing the COVID situation, balancing all aspects any biomedical lab in charge would actually face. And I had an opportunity to meet her and the team there just after pandemic, and these guys have really done phenomenal job. And good part is she has shared most of the things which will be helpful for all of us. So how we can prudently act on the situation, and the NABL audit, what you are mentioning about the in-person, and I think those were the not even like what do you say, doorstep audits. Those were to be done quickly, and you have to really get accreditation. It was a commendable job. And Arjun, of course, you have touched upon the validation aspects of course, fit for purpose is a very, what do you say? We can use the term as we want to strategize our approaches for validating and bringing really science into the system. So I think you gave a good understanding on how should we be scientific as well as comply with the current understanding of regulations, especially for the biologics. And Jen, thanks for giving the favor of advanced technology. And this was really a good blend of talks on biologics. And we talked about the traditional technologies, evolving technologies, and mass specs. So that's like, we heard also about hybrid approaches for bioanalysis of large molecules. And that's where I think, JN, you have put that LCMS icing on the cake with 7500 development. So there's a lot to learn and understand from all of you. And also there were good questions. Overall, very fascinating day today. Thanks, Deepak. Thanks all the contributors.
Thank you. Thanks a lot to both the moderators and all the three speakers. Once again, Aparna, Arjun, Darshana, and JM, thanks a lot. And looking forward to connect with you all soon and have you again in person for next year. So officially we conclude the webinar. Thank you, Aparna. Thank you, Arjun. And
Thanks,
would
Deepak.
request thank you, JM. We'd request attendees and the organizing committee members to stay back so that we can go for the draw. Thank you. Thanks, everyone.